The vector becomes infective to the vertebrate host only after elapse of certain time during when the parasite undergoes development and multiply. That means ଯଦି ଭେକ୍ଟରଟି ତାର ପାଇଗଲା ଗଟରେ ଆସିଗଲା ଟ୍ରାଇପାନୋସୋମା କିନ୍ତୁ ଗଟରେ ଆସିଲା ପରେ there should elapse certain time जो period रे it will multiply, it will change its structure and invasiveness infectiveness टा ଆସିଯିବ ସେ parasite ରେ. So the vector becomes infective only after elapse of certain time, after the vector has inoculated itself with the parasite. Okay, with the parasite. तापर जाइके ना immediately से blood milk कोठे आसिला आ गोटे healthy human को ये कला सेटा infection हबो नाही, because unless and until it develops in the gut of the vector, it will not be infective. So two modes of development inside the vector. गोटे होची in case of African trypanosomiasis, they develop and multiply in the midgut, okay, of the tsetse fly, that is the Glossina palpalis. So TSC, it is pronounced as tsetse fly. They develop and multiply in the midgut. Transmission occurs by the inoculation of parasite through the bite of the insect. Another station for multiplication, as in case of the T. cruzi, Trypanosoma cruzi, the agent for the Chagas disease, the ingested trypanomastigotes, they develop in the gut of the vector, which is a bug, kissing bug, it is generally called, and the transmission occurs not through the bite of the bug, rather, it is transmitted through the fecal matter of the bug where the parasites are present. Now, many parasites, many species of trypanosoma, they affect the human strain. It is Gambians, Rhodians, and then Trypanosoma brucei, and so on. Now, coming to there are other strains which infect the animals also, and these animal strain uh, are Trypanosoma brucei, Evansi, Equiperdum, Lewisi. These are the different. Now, it causes sleeping sickness or trypanosomiasis. It exists in two forms, that is promastigote and amastigote forms. They pass their life cycle, complete their life cycle in two host, vertebrate and the insect vector, that is, you know, uh, tsetse fly, it is called, Glossina palpalis. As I've told you, vector becomes infective only after the parasite undergoes some development and multiplication. The parasite has to multiply within the gut of the vector. Nohele, it will not become infective. It will not attain the invasive or infective stage. In case of Trypanosoma brucei subgroup, the ingested trimastigotes develop and multiply in the midgut of the tsetse fly, and in the other, as I've told you, in the bug, that is the kissing bug, it it multiplies, but it is transmitted through the fecal matter. Now, here is just the slide is shown here. It shows about the life cycle of the Trypanosoma cruzi, and this passes the vector huchi, the kissing bug. You can see how in the invertebrate stage the fecal matter are released, and the fecal matter, by chance, it contaminates the water surrounding the human being, or however, by some means, if the human being is exposed such contaminated water where the kissing bug has defecated, then there is a greater chance of the human being to become infected. Now, this is the morphology of the trypanosoma. You see how surface re jo flagellum is attached, flagellum ta free nahi, there occurs a membrane between the axoneme of the flagellum and the body of the parasite. So you can see here, this is the undulating membrane, and there is a prominent nucleus present. There is a the flagella arises from a basal granule known as the blepharoplast, like in case of all other cilia and the flagella. It is covered by a pellicle. There is a short body, a structure called the kinetoplast, and just beneath the kinetoplast, the uh, flagellum arises, and it goes beyond the, okay, it goes beyond certain part of the anterior part of the body. It emerges from the anterior part of the body. Now, mode of infection. The infection of this trypanosoma, it is generally in the human being, it is transmitted by Glossina palpalis or the tsetse fly, and both genders transmit the disease. That means male tsetse fly as well as female tsetse fly, सेठी gender discrimination nahi, like in that case of malaria parasite, जो तेरे female anopheles mosquito are only responsible for the transmission of malaria disease. Now here, both the genders, both male and female Glossina palpalis, they are responsible. They can transmit the disease through the bite, and the bite occurs, unlike that of the mosquito, it occurs in the daylight hours. So apart from the bite by the carriers, which is a tsetse fly, the transmission, the other modes of transmission includes mother to child through the placenta, then mechanical transmission through by other blood sucking. Other blood sucking insect is also possible, but it is not assessed, it is not um, evidenced. Another is accidental infection in laboratories, pricks with contaminated needle. They may also, and sexual contact uh, is uh, proposed, but such transmission has been reported, not, but it is not clearly established. Now, coming to the life cycle, that is the most. Hello.
ପାର୍ସଲ କେଣୁ ପକେଇଛି କି ଆଣି କେତେ ପ୍ରୋଜେକ୍ଟ କରି ରଖୁଛୁ ମାଡ ଭଲ ଅଛି ଭଲ ଅଛି ହ୍ୟାଲୋ
माइक 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 बंद करंदो, माइक बंद करंदो, क्लास चलची।
Okay,
Sir, please.
I think accidentally they have failed to switch off the mic. So I request all. So if you have any, please mute your mic, and then you go for your core works or anything necessary you feel. We get disturbed. Okay, anyway. Now, life cycle, it is most important for all the parasites. This topic is parasitology. We have to study about the life cycle. It is necessary. Now, life cycle of trypanosoma, that is, it is a digenetic parasite, completes its entire life cycle in two hosts, primary host being the man, and the secondary host or the intermediate host is the tsetse fly or the Glossina palpalis. Now, in the man, it is not a, it is intercellular parasite, not intracellular, unlike that of the plasmodium. Yes, blood cell bhitere roheni ki kaun si cell bhitere rohe nahi. Amidst the cell, in the matrix, the blood is a connective tissue. Tara matrix hochi plasma, the plasma re roibo. So when we take a film of the blood amidst the RBC and other cells, you find the trypanosoma, which I have shown you in my earlier slides. In the human, it lives in the blood and body fluids also, like lymph and cerebrospinal fluid. So I have explained you what is the lymph, and cerebrospinal fluid is the fluid which is present in the ventricles or the cavities of the brain, as well as the spinal uh, chamber, spinal canal. They also inhabit this region. In human, they exist in two forms. That is a stumpy form and a long, slender form. So, long, slender form of the trimestic goat is the actively dividing form. They only divide through longitudinal binary fission, and the structure is a typical elongated structure allowing efficient movement within the bloodstream with the help of the flagella it bears. And the short, thick, stumpy form, it is short in nature, stumpy, that means almost globular shape. They are broader and may have attenuated or absent flagellum. What is attenuated? Flagellum is absent, okay, we understand. Attenuated means the flagellum, if at all, it bears a small extension and it is not movable. It does not function. That is called attenuated or absent flagellum, allows them to become short, thick, and stumpy and non dividing. They will never divide, they will not undergo binary fission. They represent a stage in the life cycle where the parasite prepares for transmission to the tsetse fly. So when this parasite uh, has to enter into the body of the intermediate host, it will transform itself into the short stumpy form from the long slender form. Now this intermediate form, uh, this form has characteristics. So there is a stage. So you can say it is an imaginary stage, that an intermediate state which is in between long slender and stumpy. So they immediately, though they don't transform, rather gradually they transform from a long slender into the stumpy forms. In the vector, that is the tsetse fly, it exists in the procyclic trypomastigote form in the midgut of the vector, and in the form epimastigote or metacyclic forms in the salivary gland. As I have told you, after the ingestion of the blood meal, where the trypanosome is present, then there should be some time elapse elapsation of some time, where the in the midgut some development will occur, and after the development and multiplication, they will move to the salivary glands, where they can infect another human being or another vertebrate host. So in the midgut, they are in the form of procyclic trypomastigote, and in the salivary gland, they are present as metacyclic form. Now the life cycle here, you can see how the changes in the bloodstream when they have infected the human being. In the bloodstream, they transform this uh, metacyclic trypomastigote form. They get transformed into the long elongated form, proliferating. Proliferation means they undergo division, they undergo binary fission, and after a repeated cycle of this infection, they again some of the parasite, they stop dividing and they become non-proliferating. They don't divide, they become short and stumpy with attenuated flagellum, waiting to be taken up by the tsetse fly along with the blood meal. ଯେତେବେଳେ ସେଟ୍ସି ଫ୍ଲାଏ ବଡିକୁ ଯିବ, ପ୍ରଥମେ ତାର ମିଡଗଟକୁ ଯିବ, ତାର ଇଣ୍ଟେଷ୍ଟାଇନକୁ ଯିବ, ତାର ଡାଇଜେଷ୍ଟିଭ ସିଷ୍ଟମକୁ ଯିବ। ସେ ଡାଇଜେଷ୍ଟିଭ ସିଷ୍ଟମରେ କିଛି ମଲ୍ଟିପ୍ଲିକେସନ ହବ। ଡାଇଜେଷ୍ଟିଭ ସିଷ୍ଟମରେ ସେଇ ମଲ୍ଟିପ୍ଲିକେସନ ଇଜ ଥ୍ରୁ ବାଇନାରୀ ଫିସନ, and they, in this stage, it is called procyclic trimastigode form. Then only they will convert themselves into an intermediate stage called the epimastigode, then finally transformed into metacyclic. A metacyclic stage re, they enter into the salivary glands to ready for the infection to another human being. So this is a complete life cycle. Same thing. Um, you can see a number one here, the test. tsetse fly, it bites the human being, and the parasite is taken along with the blood meal, moves into the gut. Then here it bites, releases the trypanosoma into the human body from the salivary glands. Because it has to, when it is taking blood meal, it has to release anticoagulant to prevent the clotting of the blood. Say anticoagulant ta tar salivary gland ru bahare, say salivary gland re thiba trypanosoma guriko moniso blood ku palai ase. So here it enters into the bloodstream, or it may reach the body fluids including lymph and spinal fluid, and immediately they start replicating by means of binary fission. Binary elongated binary fission, longitudinal binary fission. You can see here in this point how how the fission occurs from the anterior towards the posterior side. So anterior side is the region where the elongation or extension of the flagella occurs. So entire life cycle is represented by the all are extracellular. Remember, there is no intracellular stage. In the third stage, the tsetse fly becomes infected with bloodstream trypanomastigotes when taking a blood meal on an infected host. So here, when the tsetse fly, it bites the human being for blood meal. Now, both dita jaka କ୍ଷେତ୍ରରେ ରିଲିଜ କରିବା ପାଇଁ tsetse fly bite କରୁଛି, ପ୍ଲସ୍ ଯଦି ଯେତେବେଳେ ବାଇଟ୍ ବ୍ଲଡ୍ ନଉଛି ଇନଫେକ୍ଟେଡ୍ ପର୍ସନରୁ ବ୍ଲଡ୍ ନଉଛି, ସେ ମଧ୍ୟ ଗୋଟେ ଷ୍ଟେଜରେ ସେ ଟ୍ରାଇପାନୋସୋମାକୁ ସେ ଗ୍ରହଣ କରୁଛି. So trypanosoma is not necessary for the insect to survive. Rather, evolution is a host-parasite relationship. So it is a means by which this parasite will complete its life cycle only when it is taken up by the tsetse fly. So tsetse fly bhitere puri midcutre transformation hochi, division hochi, transformation hochi. They build up, they increase their number, and again ready to be infected to the next fresh person when it is loaded in the salivary glands. Now, um, there are three stages we can find in the sufferings. So, first stage is the hemolymphatic stage. Now, this stage, the warmth of the human body or the vertebrate body, they they provide a medium, they provide a platform for this long and slender form um, to be transformed, and they multiply in the blood. So the worm transform them into long and slender form from the stumpy form jota asila tsetse fly ru ମଣିଷର ବା vertebrate ର ଯୋଉ warm blooded animal, ସେଇ warm blooded condition ତାକୁ ଫେଭର କରୁଛି elongate ହବା ପାଇଁ ଆଉ divide କରିବା ପାଇଁ through longitudinal binary fission. ତାପରେ this may be carried to other sites. ନିଶ୍ଚିତ ଭାବରେ ବ୍ଲଡ୍ ବ୍ରେନ୍ ବ୍ୟାରିୟରକୁ କ୍ରସ୍ କରିକି ସିଏନ୍ଏସ୍ କୁ ଯାଇପାରିବ, ଲିମ୍ଫ ଷ୍ଟ୍ରକ୍ଚରକୁ ଯାଇପାରିବ. This may be carried to other sites of the body and reach other body fluids, spinal fluid, where they continue replication by binary fission. Some remain as long and slender form, while others take the form of short and stumpy type of mastigotes. କାହିଁକି କାହିଁକି କଣ୍ଟିନ୍ୟୁ କରନ୍ତି ନାହିଁ? ନା ସେଇଠି ହୋଷ୍ଟ ପାରାସାଇଟ୍ ରିଲେସନସିପରେ ସେ ରହିଛି. ଗୋଟିଏ ଗୋଟିଏ ଲୋଡ୍, ଗୋଟେ ଫିକ୍ସଡ ଲୋଡ୍ ପରେ they will not allow the host to die. ସେ ଜାଣିଛନ୍ତି ଯେ if we continue, then there will be a much parasitic load on the host body that will render the host to die, and if the host die, we will also die. ତ ସେଇଟା ଗୋଟେ ଇଏ ରହିଛି, ପ୍ରକ୍ରିୟା ରହିଛି. They will stop. Some they stop dividing and they enter into the short and stumpy form. Second stage hochi neurological menin encephalic stage. The long and slender forms sometimes it cross the blood brain barrier. इटी blood brain barrier माने होची कौन गोटे जो ब्रेन को जो ब्लड कैपिलरीज जाइची, especially these blood capillaries of the brain, they are they are highly specific to allow certain substances only to pass, and not they will not allow, they are selective in nature. But these parasites, they are capable to cross this blood barrier and enter into the brain tissue, so infect the central nervous system. And this stage is important. जो कथा में कहूचे जे trypanosoma or sleeping sickness, it is this condition, they is stage 2 रे they come nearer to the brain, and they impact on the brain causes the person suffering or with the trypanosoma to have a sleepy attitude. So, see term asila African sleeping sickness. So the peculiar phenomenon of disturbance of the sleep cycle, तार sleep cycle टा सेटा disturbed हे जाउची, जोटा हमर body रे sleep cycle होची, sleep awake cycle होची, सेटा totally disturbed हे जाउची, आउ से सदा in spite of different times the sleep cycle is changed. से सबले मांदा रहउतिबे, निद लागुतिबो, lethargic लागुतिबो, which gave the name as sleeping sickness. तापर stage 3 आसिला, एटा होची setsy fly stage. Setsy fly stage होची the stage at which it will now become ready to be infected by the to be sucked in by the tsetse fly. ତେଣୁ long and slender form କିଛିଟା ସେମିତି ରହିଥିବା, ଆଉ କିଛିଟା they become short and stumpy, ଆଉ short and stumpy ହେଇଗଲା ପରେ ଆଉ divide କରିବେ ନାହିଁ, blood ରେ ସେମିତି ଥିବ। ଅକସ୍ମାତ accidentally ଯଦି tsetse fly ଆଉ ଗୋଟେ ଆସିକି କାମୁଡ଼ିବ, ତ କାମୁଡ଼ିବ କ'ଣ ପାଇଁ? ବ୍ଲଡ୍ ନବା ପାଇଁ। ତ ସେଇ ବ୍ଲଡ୍ ସହିତ ସେ ଇଏ ଗୁଡ଼ାକ ଚାଲିଯିବ, କୋଉଟା ଯୋଉ short and stumpy form. Trimastigotes, while taking a blood meal from the infected host, the parasite transformed into तार gut रे जो tsetse fly ର mid gut रे procyclic अवस्था को चली जीवो, then it will transform into epimastigotes, ଆଉ epimastigote stage रे they will reach the salivary glands and continue salivary gland रे पुणी multiply करिवे, ଆଉ finally they reach the stage of metacyclic tripastigotes, by which they are ready to infect a fresh person. Now it देखिबो, देखो ये गोटे से ये अल्सरेशन हे जाइची, अल्सरेशन माने से रीजन रे जतबेले सेट्सी फ्लाई कामुडीची, तो सेटा केमती व्हाइटिश हे जाइची, you can see here the ulcer, and sometimes the typical fine spotted and khimiri body pink rash in the abdomen. These are the symptoms you can see find here. That is called the ulcer को कहा जाउची trypanosomal ulcer and this ulcer एटा जति तारो टिके सेठी ब्रेक हे जिवो ना, and it will allow secondary infection. तापर secondary infection आरंभ हे गला माने it is very difficult for to subside the region, to subside the ulcer. ଆଉ hemolymphatic phase होची कोन, bouts of fever आसीबो, मुंड बिंदिबो, fever can last from a day to a week, separated by intervals of a few days to a month or longer. So jara आसीबो, एगुडी होची particular symptoms. ଆଉ neurological symptoms तुम कहिली जो sleep awake cycle टा disturb हवा जोगु, they always the sleep wake disturbance lead to giving the disease as sleeping sickness. So they experience sleep in the daytime also. सदाबले निद लागिबो, सदा नाइट टाइम रहिबो, एंड नै डे टाइम रे स्लीप वेक साइकिल टा डिस्टर्ब हे गला पा, आउ नाइट टाइम रे पीरियड्स ऑफ इंसोम्निया, सो इरिटेशन हबो, डिजीनेस हबो, पुअर कोऑर्डिनेशन, तो ब्रेन को अफेक्ट कला ने पुअर कोऑर्डिनेशन माने कोन कहउछन्ती किछि जानि परिबेनी, डिजीनेस ये रहिबो, बाउल चाली हेबे, सो इटा होची इम्प्रॉपर कोऑर्डिनेशन, गोटे कथा भाबुथिबो किंतु कहिबो आ गोटे कथा, ମାନେ ଜେନେରାଲି ସବୁ କ୍ଷେତ୍ରରେ ଏଇଟା ଯେ ସେଟ୍ସିଫ୍ଲାଇ ଏ ଯୋଉ ଟ୍ରାଇପାନୋସୋମା ଇନଫେକ୍ସନ ସେ one of the reason is that lack of coordination. Our coordination is always mediated by the brain. So coordination ଗୋଟେ example କହୁଛି ଯୋଉ suppose you are thinking something and you are penning it same thinking ସେଟା ହଉଛି coordination effectively. So your writing it involves muscles so ତମର thinking so ଏଟାକୁ ଇଏ କରାଯାଏ ସେଇ coordination ଏଥିରେ जो ट्राइपानोसोमा इन्फेक्शन रे देखा जाए नहीं नाउ डायग्नोसिस होची यूजुअली जे ब्लड फिल्म ता होची बेस्ट डायग्नोसिस टेक्निक प्राय जा हमरो अंडर डेवलप्ड कंट्रीज रे ब्लड प्रोफाइल एनालिसिस करा जाउची सो टू टाइप्स ऑफ ब्लड प्रोफाइल एनालिसिस हमरो वेट प्रिपरेशन रे तो वेट प्रिपरेशन कोन होची ना व्हेन the blood is not right so you can find the motile phases of the trypanosome gambians in the sample and smear ताको हमें कहबा ड्राई फेज सेतिर कोन जेम्सा स्टेन रे जोटा मो स्लाइड देखैतिली सेतिर you can find the presence of this trypanosome amidst the blood cells okay नेक्स्ट सेरोलॉजिकल असेसमेंट सेटा होची जेतबे सेरोलॉजी रे हमें जानिबा तो इम्यूनोजेनिक सो प्रेजेंस ऑफ एंटीबॉडी अगेंस्ट दैट पैरासाइट तो से सेरोलॉजिकल असेसमेंट हमें करि परिबा दैट इज कैट जो कार्ड एग्लूटिनेशन टेस्ट फॉर ट्राइपैनोसोमियासिस स्पेसिफिक सेटा जो सेरोलॉजिकल तो ब्लड रो सेरम टा बाहर करी करी ब्लड रो तापरे ताको देखिबे जे वेदर एंटीबॉडी इज प्रेजेंट और नॉट अगेंस्ट so that is now immunity body ro always all human body or animal body ସେମାନଙ୍କର ଗୋଟେ immune system ରହିଛି ରୋଗ ପ୍ରତିଷେଧକ ଶକ୍ତି ରହିଛି so immunity ମଧ୍ୟ ସବୁ organism ପାଇଁ ରହିଛି so similarly in case of trypanosoma also we have immunity against so human innate immunity against most African trypanosome is mediated by the toxic serum HDL so innate immunity means the immunity which we have inherited from birth तो ସେଥିରେ ହେଉଛି toxic serum HDL trypanosome lytic factor so ଏଇ trypanosome lytic factor ତାମାନେ they are present and they will cause a lysis of the trypanosome okay lysis of this trypanosome so এই जो HDL ରହିଛି human innate human innate immunity HDL based it is called the factor is called trypanosome lytic factor एथिरे proteins रहिछि and other compounds रहिछि which are responsible for the lysis of this animal जदि एनिमल एक्सेस करूची ता माने तारो निश्चित भाबरे मेकानिज्म रहिछि टू काउंटरएक्ट दिस trypanosome lytic factor now then trypano tolerance it is a trait in human that confers the capacity to survive and remain productive upon trypanosome so what a tolerance রয়েছে tolerance রয়েছে মানে certain human being they will tolerate so say capacity is related to a character so that a trypano tolerance character so this character present in these persons who inhabit the region where trypanosome are present so they have evolved this trait say ପ୍ରକୃତିଟା ସେମାନଙ୍କର ଇଭଲ୍ଭ ହେଇଯାଇଛି ଯାହାଦ୍ୱାରା କି even if the trypanosome is present in their body they can still survive and remain productive upon trypanosome infection then there are other co-infection of parasites co-infection of parasite con now co-infection of parasite strains can also play a major role in attenuated disease progression and suppression related to the jodi simultaneously सेट्सी फ्लाई मल्टीपल बाइट रे बहु प्रजातिर ट्राइपानोसोम को बॉडी को छाड़ी दैछन्ती ताको हमें कहबा को इन्फेक्शन ऑफ पैरासाइट से क्षेत्र रे ऑल विल टेंड टू मल्टीप्लाई एंड दे विल इंटरफियर विथ ईच अदर कारण जेनेटिकली दे आर डिफरेंट so such condition naturally semane kon karibe they will suppress or disease progression को semane attenuate karibe प्रभाव हीन करिदेबे so they play a major so the load of the parasite due to multiple strain of the parasite causes attenuation of the disease progression and they suppress the disease. Then similarly immune evasion ରହିଛି ନହେଲେ parasite ଯଦି evasion mechanism ରହିବ ନାହିଁ ତାହେଲେ parasite ତ କେବେ ହେଲେ they will uh, they will not be able to survive in the body. Just a minute. One minute. Okay. So diagnosis, blood diagnosis, so the immunity, tolerance, as I have told you, trypano tolerance, co-infection of parasites, serological study diagnosis kara jai paribo. Now coming to the prophylaxis. So prophylaxis is the prevention and control. How you can prevent? Simply, uh, s- all the wild animals on which a tsetse fly feeds, slaughtering the allow kora jeni. Don't allow the region, or rather we say, don't allow the medium to sustain, which will act as a breeding ground for these tsetse fly or the Glossina palpalis. So taku kuhajae killing, reservoir killing, you kill. Then mechanical trapping, simply uh, it is effectively managed using simple inexpensive traps, often using electric blue cloth or whatever. Mechanical trapping means you don't allow the tsetse fly to bite. You just trap them and get rid of them. ताको बाहर कर दबो. Then chemical pesticides, so inorganic metal based pesticides, they also can be effective in controlling these insect. You kill this insect. Wild animal reservoir killing, as we have already then sterile insect technique, if you এটা এই যে sterile insect technique টা এটা ବହୁତ widely ବର୍ତ୍ତମାନ acceptable ହେଇଯାଉଛି. If we render them the vectors sterile ଆମେ ଯଦି କରିଦବା ସେମାନେ ତ reproduce କରିପାରିବେନି. Their population will gradually decrease and they will finally be eradicated from this globe. So সেটা হচ্ছে sterile insect technique. Ionizing radiation like gamma ray, x-ray to mass produce sterilized male flies. So male flies sterilize ହେଇଗଲେ female flies ଆଉ reproduce କରିପାରିବେନି, ତାଙ୍କ population କମିବନି. So same technique ସେମାନେ ଚିନ୍ତା କରୁଛନ୍ତି to employ in case of the mosquito also. So prevention and control, preventive measures. You preventive means get rid of yourself from the bite of the tsetse fly, wearing long sleeved shirts and pants while going outside. Then insect repellent, ଯାହା ଦ୍ୱାରା ସେଇ ମସ୍କିଟୋରେ ଯାହା ଆମେ ଇଏ କରିଥିଲେ ସବୁ ମଶାରୀ ପିନ୍ଧିକି ଶୋଇବା, ଡେ ଟାଇମରେ ବାହାରକୁ ଯିବାନି. ସେମାନେ ଡେ ବାଇଟରେ ନା, ଡେ ଟାଇମରେ ବାହାରକୁ ଗଲେ ଫୁଲ ପିନ୍ଧିକି ଯିବା ଆଉ insect repellent ଲଗେଇଥିବା ବଡିରେ, so they will not be attracted to bite. Then ତାଙ୍କର ଯୋଉ ଯୋଉ ଭ୍ୟାକ୍ସିନେସନ ବା ଡ୍ରଗ ତିଆରି ରହୁଛି, ଡ୍ରଗ ଟାର୍ଗେଟ ମୁଁ କାଲି କହିଥିଲି. ଡ୍ରଗ ଟାର୍ଗେଟ ହଉଛି କ'ଣ, ତାଙ୍କର ଯୋଉ ୱିକ୍ ପଏଣ୍ଟ, ଗୋଟାଏ ଗୋଟାଏ ପାରାସାଇଟ୍ ବା ଗୋଟିଏ ଏନି ଇନଫେକ୍ଟିଭ ପାଥୋଜେନର ୱିକ୍ ପଏଣ୍ଟଟାକୁ ନୋଟିସ୍ କରିକି ସେଇ ୱିକ୍ ପଏଣ୍ଟ ବିରୁଦ୍ଧରେ ଆମେ ଡ୍ରଗ ବା ମଲିକ୍ୟୁଲ ତିଆରି କଲେ ତାକୁ କୁହାଯାଏ ଡ୍ରଗ ଟାର୍ଗେଟ୍ସ. Okay, drug targets. Suppose the parasite, suppose tsetse fly, ସେମାନଙ୍କର salivary gland ରେ ଯୋଉ enzyme ରହିଛି, that will facilitate the living state of the trypanosoma. So if we can genetically modify, ତ ସେ enzyme ଟା produce ହବନି. enzyme produce ନହେଲେ they will not allow the to for a platform for the breeding of this in their salivary gland. এটা হচ্ছে ଆମର ଯୋଉ target. तो सब एनिमल रो समान नो साइंटिस्ट से माने दे विल ब्रिंग अबाउट दे कोटा को आमे टार्गेट करिबा गोटे वीक पॉइंट ऑफ द ऑर्गेनिज्म सब बोले मन रखिबो वीक पॉइंट ऑफ द ऑर्गेनिज्म इज ऑलवेज टेकन इनटू कंसीडरेशन एंड द इजीएस्ट वे and this weak point mane kaha कहबा without which the organism cannot multiply hala it will never favor the existence of a parasite सेटा होची तारो weak point. Now, cure rate is a better indicator than egg reduction rate for assessment the efficiency of anti-parasitic drug agent. Suppose ଆମେ କହିବା ଯେ ନାଇଁ ଆମେ ତାର egg ଟାକୁ number of eggs ସେ ଯେତିକି produce କରୁଛି ଆମେ ତାକୁ କମେଇ ଦେବା. No, egg ତ produce ହଉଛି ନା even a single egg ଯଦି infection ହେଲା ସେ ରହିଲା ତାର population ରେ ସେଟା ରହିଲା. So ସବୁଠାରୁ main ହଉଛି cure rate. How will you cure? Cure means the affected person is rendered healthy. Then egg reduction rate is an estimated post-treatment account with a comparison with the pre-treatment load for per common unit of the so specific drug and treatment course depend on the type of infection. So where you decrease the number of eggs laying capacity or you will cure, it depends upon the species. So drugs available rahichi pentamidine, suramine, these are the drugs of the first choice. First stage रे तापरे जदि aggravate ही जाइथिबो first stage aggravate माने the multiply करिकि बहुत गुडे ता संख्या so melar soprol then nifurtimox these are different molecules सब ना गुडी कर रहिछि. Now we'll move on to the vaccine ମଧ୍ୟ ଇଏ ରହିଛି so କୌଣସି କ୍ଷେତ୍ରରେ କୌଣସି full fledged vaccine produce ହେଇପାରିନି ଯେମିତି vaccine also strategy ତାର ମଧ୍ୟ ଗୋଟେ target parasite ରହିଛି targeting the parasite targeting the weak point of the parasite. So for example flagellar pocket. So flagellar pocket মানে the region from which the flagella of the trypanosoma arises. A common architectural feature of trypanosoma is the flagellar pocket. FP taku kaje. It is formed by an invagination of the membrane at the base of the flagellum. It is readily involved in exocytotic and endocytotic processes, cell division, and polarity. Remember, I again repeat, flagellar pocket is necessary for exocytic and endocytic processes. মানে endocytosis ହେଇପାରିବ exocytosis cell division and polarity protein trafficking and more important so cell division ମଧ୍ୟ se control करे. These features make FP antigens attractive candidates. ता माने if we make the FP effectiveness तो से multiply करिबो नाही parasite load body रे बढ़िबो नाही so we can target. So it is target possible targeting the parasite targeting a particular region of the parasite. Then tankar jo actin tubulin jo protein rahichi cytoskeleton because they are capable to move the movement ପାଇଁ ନିଶ୍ଚିତ ଭାବରେ molecules ରହିଛି cytoskeleton ରହିଛି actin tubulin ରହିଛି they are also targeted for the development of the vaccine କେମିତି actin is known to play a crucial role in bloodstream forms not only due to its involvement in cell division but movement and morphology also. Actin cell division माने जो spindle fibers गुडी को formation होची तारो role रहिची as well as in movement. If the vaccine is targeted against the development of actin or weakening the actin or tubulin, they will not divide. They will not be able to move. So এটা হচ্ছে targeting the particular region of a parasite for the development of the vaccine. So prevention and control to mo କହିଦେଲି. Now we'll move on to the next that is the cystosoma hematobiome or this is another peculiar parasite of the blood parasite of the body. This parasite cystosoma hematobiome it causes the disease schistosomiasis. It is a blood fluke belonging to class Trematoda. Schistosomiasis होची तारो disease ना. So a slide रे छोटा देखा जाए ची, you can see here, it is a combination of both male and female, you know. So ये जो thin जो thread like structure, that is the female, it resides in the gynecophoral canal of the thicker male. So male or female, they cannot be separated. Okay, ସେମାନେ separate ହୁଅନ୍ତି ନାହିଁ, ଏକାଠି ରହନ୍ତି. That is the peculiarity of this parasite. Okay, of this parasite. So then generally this এই जो সিস্টোসোমিয়াসিস, এটা হচ্ছে পানি তার প্যারাসাইটর গোটে লার্ভাল স্টেজ, ইট ইজ প্রেজেন্ট ইন দি ওয়াটার। তো ଯେଉଁମାନେ the persons who have a very unhygienic, or they dwell in a contaminated water, or they are in the rural community, poor community, ଯୋଉଠି ଅଧିକ density ରେ ଲୋକ ରହୁଛନ୍ତି, and they are involved in fishing activities, or they are involved in washing in the infected ponds. ତ infected water ସହିତ ଯୋଉମାନେ କାମ କରୁଛନ୍ତି, ସେମାନଙ୍କ କ୍ଷେତ୍ରରେ ଏ ପ୍ରକାର ଏ ଯୋଉ ସିଷ୍ଟୋସୋମିଆସିସଟା generally ଦେଖାଯାଏ। So basically the fishermen who wade in contaminated lakes and streams, because their larval stage hochi infected stage. In streams, they carry the risk of schistosomiasis, and it is generally found in the population which are economically very poor, and they are also poor in education also. ସେଇଥିପାଇଁ awareness ର ତାଙ୍କ ଉପରେ କୌଣସି ପ୍ରକାର ପ୍ରଭାବ ପଡ଼େ ନାହିଁ। If they are not educated, they will not understand what is good or what is bad. Characters होची एठी जो मेल animal टी देखुछ जो thick रहुची, the male is around it may be more than 2 centimeter long, more than. तो मेल बॉडी रे गोटे वेंट्रल ग्रूव रहिची, ଆଉ से ग्रूव ର ନା होची गाइनेकोफोरल कैनाल। ओके, गाइनेकोफोरल कैनाल। এই গাইনোকোফোরাল ক্যানাল ମଧ୍ୟରେ ସେମାନେ ରହନ୍ତି। କିଏ? ଫିମେଲ ବଡି ରହନ୍ତି। ଫିମେଲ ବଡି is around 2.5 centimeter long, and it does not exactly fit into the body of the male, but they are thinner than the male, and they reside within the gynecophoral canal of the male, ଯୋଉଥିପାଇଁ ତା ନାଁ ହେଲା ସିଷ୍ଟୋସୋମ। ସିଷ୍ଟୋ ଗ୍ରୀକ ୱାର୍ଡରେ ସିଷ୍ଟୋର ଅର୍ଥ ହଉଛି ସ୍ପ୍ଲିଟ୍, ତା ମାନେ ଡିଭିଜନ ହେଇଥିବ, ଆଉ ସିଷ୍ଟୋସୋମା ମାନେ ହଉଛି ବଡି। ବଡି ଇଜ ସ୍ପ୍ଲିଟେଡ, ବଡିରେ ଗୋଟେ ଗ୍ରୁଭ ରହିଛି, ଆଉ ସେଇ ଗ୍ରୁଭର ନାଁ ହଉଛି ଗାଇନୋକୋଫୋରାଲ ଗ୍ରୁଭ, ଗାଇନୋକୋଫୋରାଲ କ୍ୟାନାଲ, and this groove allows for the dwelling by the female animal of the cystosoma. Now, right ରେ ଏଗୁଡ଼ା ସବୁ different species ର cystosoma hematobiome ସେଟା ରହିଛି, ଏଠି ଜାପନିକମ୍ ରହିଛି, hematobiome ରହିଛି, mansoni ରହିଛି. These are the different eggs, ତାର structure difference ରହିଛି. You can see here the spine ର position ଆଉ spine ର sharpness, it determines କୋଉ ପ୍ରଜାତିର କୋଉ species ର ଏ parasite ରହିଛି. It is generally determined by the type of spine that the eggs have on their body. Now, you must be aware of the Cercaria larva. ଆମେ Cercaria larva ପଢିଥିବ liver fluke ରେ, Miracidium larva, Sporocyst larva, Cercaria, Metacercaria. So here also there is a larval stage known as the Cercaria larva of cystosoma. Now the life cycle of this particular parasite, जोटाको ଆମେ cystosoma कहुचे, it was the first cycle out of 3 primary human cystosome species, that is Cystosoma japonicum, which has been studied completely out of the three species there. Now, the key differences uh, among the three type of life cycles followed by the three species of the cystosoma depends upon the intermediate host, which is a snail. ମନେ ରଖିବା ଆମେ ଫ୍ୟାସିଓଲା ହେପାଟିକାରେ ମଧ୍ୟ ଗୋଟେ ଇଣ୍ଟରମିଡିଏଟ୍ ହୋଷ୍ଟ ରହିଥିଲା, ଯୋଉ ସ୍ନେଲ ରହିଥିଲା, ଏଥିରେ ମଧ୍ୟ ସ୍ନେଲ ରହିଛି। ନାଉ ଦି species of the snail, same species is not taken into consideration by all the three species of the cystosoma. କାରଣ ଗୋଟିଏ ସ୍ପିସିସ୍ ହେଇଗଲେ ଡିଫରେଣ୍ଟ ସ୍ପିସିସ୍ର ସେମାନଙ୍କ ରିକ୍ୱାରମେଣ୍ଟ ଅଲଗା ରହିଥିବ, ସୋ ଦେ ଫଲୋ ଡିଫରେଣ୍ଟ, ଦେ go through different snail species. So maybe evolutionary ray, it is my personal view, they they have made them compelled the snail species to evolve into a new species for their adaptation. Okay, for their adaptation, which I believe. So different snail species must have arisen by this cystosoma. They have now female lay fertilized eggs in the portal venules and perivesical systems. The fertilized females eggs pass out of the body through the fecal matter. Okay, through the fecal matter. The females lay eggs in the portal venule. So portal system means the blood circulation. Generally, the blood flows from. It is collected from body organs and immediately it goes to the heart. But there are certain circulation where the blood collected from one organ passes into another organ before going to the heart. Such circulation type is called portal circulation. तो एमाने portal circulation रे तांकर portal venules रे female माने से माने ये करिवे release करिवे तांकर जो eggs गुड़िक, and ultimately portal circulation रे it they meet with the uh, intestine, and ultimately they are released into the fecal matter. Okay, so human being are the reservoirs for the disease. Monisha hochi the definitive host, and intermediate host hochi the snail. Now, when they are released into the along with the fecal matter, and the it reaches the fresh water. It is fresh water means not the clean water. It is fresh water means it is not a marine water, so they cannot. So this fresh water means it may be contaminated water. Okay, so fresh water ଯେତେବେଳେ release of, once they again reach the fresh water, they hatch into the eggs hatch into the free living non-feeding ciliated larval stage, same like that of the Fasciola hepatica. The first larval stage is the Miracidium larva, जोतिर की entire body is covered with cilia, which allows them to swim effectively in the water medium. So this presence of cilia ତାଙ୍କୁ allow करे to swim in the water, and during the swimming, due to chemotaxis, they will be oriented towards the specific species of the snail जोटिक ସେମାନେ enter କରିକି they will complete their life cycle. So they infect the intermediate snail host depending upon the species of the cystosoma. Now here जो सरकारिया larva एठी चित्र ଦେଇଛି. Now in the snail से development କରିବ, ଯେମିତି Miracidium larva in the snail ରେ development କରିବ କଲା, karan it will transform into the sporocyst. So when the Miracidium enters, the Miracidium kahinki kuhagala, because it is similar to that of the Miracidium larva of liver fluke. Entire body is covered with a cilia. So when this enters through the soft tissue of the snail ro bi entire body to covering round in snail kote operculum badi jetobale se movement କରୁଛି ବା ପାଣିରେ float କରୁଛି, soft tissue ଗୁଡ଼ିକ expose ହଉଛି, it penetrates the snail's soft tissue and eventually makes its way into the immediately the liver को पड़े, liver रे it loses its cilia. So most of the parasite, at least in the larval stage, they move into the liver because liver hଉଛି the main organ ଆଉ ସେଠି stored food ରହିଛି. It loses its cilia and other organs ମାନେ କଣ ସିଲିଆକୁ ମୁଭମେଣ୍ଟ ପାଇଁ ଯୋଉ organ ରହିବ protection ପାଇଁ ଯୋଉ organ କାରଣ when they are outside the host body they are always in the danger. So all these organs that serve as a protective measure for locomotive motion, they are all discarded. ସେଗୁଡା ସବୁ ବାହାରିଯିବ and experiences developmental changes during the period of 4 to 8 weeks. ତାରି ଭିତରେ ରହିବ 4 to 8 weeks. Finally, through certain changes, different levels of changes, the miracidium larva turns into a sporocyst, a tubular sporocyst that multiplies. Same thing in case of fasciole hepatica, it will multiply and produces daughter sporocyst. Second, ଆମେ ପଢିଥିଲେ second generation radial larva, ତ ଏଥିରେ second generation ବା daughter sporocyst, ମୂଳ sporocyst ରୁ ପୁଣି daughter sporocyst ହବ. These daughter sporocyst then they transform into our radia larva ନାହିଁ, ତାର ଲାର୍ଭା ହଉଛି ସରକାରିଆ ଲାର୍ଭା ଡାଇରେକ୍ଟଲି, କାରଣ ସରକାରିଆ କାହିଁ କୁହାଗଲା, ଆମେ ରେଡିଆ କାହିଁ କୁହାଗଲେନି ନା ଲେଭର ଫ୍ଲୋଗ୍ର ମଧ୍ୟ ସରକାରିଆ ଲାର୍ଭାରେ ତାର ଗୋଟେ ଟେଲ ରିଜନ ରହିଥିଲା. ସୋ ବାଇଫର୍କ ଟେଲ ଏଠି ରହିଛି, ଇନଫେକ୍ଟିଭ ଫଗ୍ଡ ଟେଲ, ହଁ ଫଗ୍ ଟେଲ ମିନ୍ସ ଟୁ ଡିଭିଜନ ରହିଛି. ସୋ ଫଗ୍ ଟେଲ ସରକାରିଆ ଲାର୍ଭା are released. The last larval forms which break free from the sporocyst. Sporocyst ରେ ଏଇ ସରକାରିଆ ଲାର୍ଭା ଡେଭଲପ ହେଇଗଲା. Sporocyst ରେ ଯେତେବେଳେ ଡେଭଲପ ହେଇଗଲା, sporocyst is fully loaded with the cercaria larva, ତାକୁ ଭାଙ୍ଗିଦେଲା, ତାକୁ ଫଟେଇଦେଲା, ରପଚର କରିଦେଲା and this cercaria larva they escape from the cell into the snail into the water. ଆଚ୍ଛା, କେମିତି ଆସିବ ସ୍ନେଲ ରୁ ୱାଟର ଭିତରକୁ କେମିତି ଆସିବ, ତାର ତ ଲିଭର ରହିଲା. you know snail ର both aquatic and aerial respiration ରହିଛି. So even in the water also, ଯେତେବେଳେ ସେ ଇଏ ରହିବ କଣ ୱାଟରଟା କଣ୍ଟାମିନେଟ୍ ରହିବ, ସେଠି ଅକ୍ସିଜେନ ଅଭାବ ରହିବ, they simply extend a siphon like structure. They come to the surface, extend a siphon like structure which will collect air from the atmosphere and they will utilize the air for their body metabolic activities. ତ ସେତିକି ବେଳେ ସେ ଯୋଉ ବଡିରୁ ଗୋଟେ ସାଇଫନ ଲାଇକ୍ ଷ୍ଟ୍ରକ୍ଚର ଏକ୍ସଟେଣ୍ଡ ହଉଛି, ସେଇ ବାଟ ଦେଇକିରି ସରକାରିଆ ଲାର୍ଭା ଦେ ଅଗେନ୍ କମ୍ ଆଉଟ୍ ଇନ୍ ଟୁ ଦି ୱାଟର. ତ ୱାଟରକୁ କଣ୍ଟାମିନେଟ୍ କରିଦେଲେ. କଣ୍ଟାମିନେସନ ହେଇସାରିଲା ପରେ the cercarial larval stage of the cystosoma is infective to humans. Now you can see here cercarial larva ରେ ଏଠି ଗୋଟେ oral sucker ରହିଛି. The oral sucker ଯୋଉଠି ରହିଛି ମାନେ it serves as the organ for attachment. This is the body and you have certain pre-acetabular glands. Acetabulum, remember fasciole hepatica ରେ ପଢିଥିବ acetabulum, and ventral sucker zone madhya ରହିଛି. Same thing, and these are the sensory papillae. They will sense the availability of the human being. তো সেটা সেন্সরি প্যাপিলে রয়েছে. সো এটা যদি বডি হেলা, the remaining part is called the tail, and this is the bifurcation of the tail end, that is furca. Bifurcation furca. Next, this is the life cycle of cystosoma. You can see here, this is the cercarial. Now ଏକ number ଆମେ ଯିବା, ଏକ number ଏଇଠି ରହିଛି. So in stage this stage 1, in stage 1 you can find these eggs are released into the eggs are released, they come out along with the fecal matter. Fecal matter ରେ eggs ଗୁଡ଼ିକ ଆସିଗଲା. Eggs in feces, ଏଠି ଲେଖା ହେଇଛି eggs in feces or urine, and these eggs when they reach the water they immediately hatch out and the miracidium larva comes out. So miracidium larva is completely covered with cilia. They enter and find ଏଠି ତିନି ପ୍ରକାରର ସ୍ନେଲ ଦେଇଛି, ତିନି ପ୍ରକାରର ପ୍ରଜାତିର ସିଷ୍ଟୋସୋମା. ଓକେ, ସିଷ୍ଟୋସୋମା. ସୋ ମେନଲି ମାନ୍ସୋନି ରହିଛି, ହେମାଟୋବିୟମ ରହିଛି, ଆଉ ଗୋଟେ ମଧ୍ୟ ତାର ଇଏ ରହିଛି, species ରହିଛି. So they select the type of species of the snail আর স্নেল ভিতরে দে ডেভেলপ ইনটু দি স্পোরোসিস। এটা দেখো, এটা হচ্ছে স্পোরোসিস স্টেজ। দে লুজ দি সিলিয়া। স্পোরোসিস স্টেজ রে দি এন্টায়ার স্পোরোসিস স্ট্রকচর ইজ লোডেড উইথ সরকারিয়া লার্ভা। ଆଉ ଯେତେବେଳେ ଲୋଡ୍ ଅଧିକ হবে, ସର୍କାରିଆ ଲାର୍ଭା ଦେ ରପଚର ଦି ବଡି ଅଫ ଦି ସ୍ପୋରୋସିଷ୍ଟ, କମ୍ ଆଉଟ୍ ଇନଟୁ ଦି ୱାଟର। ଏଇଟା ସବୁ ପାଣିରେ ରହିଛି। ତ ପାଣିରେ ସେମାନେ ଫ୍ରି ହେଇଗଲେ। ଓକେ, ପାଣିରେ ଫ୍ରି ହେଇଗଲେ, ଏଇଟା ସ୍ନେଲ ବଡିରୁ ବାହାରିଲା। ଓକେ, ସ୍ନେଲ ବଡିରେ ରପଚର ହେଇକିରି ସ୍ନେଲ ବଡିରୁ ବାହାରିଲା। and now it is in direct, it can directly penetrate the skin through its effective acetabulum or the sucker like structure. So basically, these Cercaria larva, it will enter the definitive host of the including human being by penetrating through the unbroken skin. Okay, through the unbroken, they will first attach by their acetabulum or sucker like structure, and they will move into the body. Cercary larvae have a tail that allows them to swim to find way in order to get entry into the vertebrate host. But once they enter the peripheral venule, the tail is cast off. So vertebrate host body को पसिगला माने तो blood circulation contact रे आसिगला, and तापरे से venule माने veins माने the blood vessel where the blood is carried to the heart. ସେଥିପାଇଁ venules ଆମେ କହୁଛେ ସବୁବେଳେ, କାରଣ ସେମାନେ ହାର୍ଟ କୁ ପଳେଇବେ। The tail is cast off, and these are called cystosomulae. This Cercaria larva without a tail, it's called cystosomulae. Cystosomulae. Parasite then travel through the lungs. ସେ ଯେତେବେଳେ vein ମାନେ ହାର୍ଟ କୁ ଗଲା, ହାର୍ଟ ମାନେ ଲଙ୍ଗସ୍ କୁ ଯିବ, ହାର୍ଟରୁ ବ୍ଲଡ୍ ଗୁଡିକ ଲଙ୍ଗସ୍ କୁ ଯିବ। କାରଣ ଭେନ୍ସ ରେ ଇମ୍ପିଓର ବ୍ଲଡ୍ ରହିଛି, ଲେସ୍ ଅକ୍ସିଜେନେଟେଡ୍। ସେମାନେ ରାଇଟ୍ ସାଇଡ୍ ଅଫ୍ ଦି ହାର୍ଟ କୁ ଯାଆନ୍ତି। ରାଇଟ୍ ସାଇଡ୍ ଅଫ୍ ଦି ହାର୍ଟ ରୁ ଇମିଡିଏଟଲି ବ୍ଲଡ୍ ଯିବ କୋଉଠି to the lungs for purification. So they reach the lungs. ସେଇଥିପାଇଁ they prefer the venules. venule ମାନେ ହଉଛି the capillary of the veins, ତାକୁ venule କୁହାଯାଏ। So they finally reach the lungs, ତାପରେ followed by heart, and finally mature in the liver. liver ରେ ତାଙ୍କର ସ୍ଥାୟୀ ବସା ସେମାନେ ସ୍ଥାପନ କରନ୍ତି। liver ଭିତରେ they exit the liver via the portal vein system and copulate in the mesenteric vein that drain the intestines or the visceral venous plexus of the urinogenital system. Yeah. So liver ରେ ରହିଥାନ୍ତି ଯୋଉ mesenteric vein, mesentery ହଉଛି କଣ, ଗୋଟେ organ, ଆମର ବଡିରେ organ ଗୁଡିକ ତା ଚାରିପଟେ ଚାଲ ଭଳି structure ରହିଛି to keep the organ in position, ତାକୁ କୁହାଯାଏ mesentery। ତ mesentery ମଧ୍ୟ cellular in origin, ସେଥିରେ ବି tissue ରହିଛି। they are also in living state, they are also supplied with blood vessel। ତ ସେଇ mesenteric vein ରେ ଏଇ ଯୋଉ male and female ତ ଏକାଠି ରହିଛନ୍ତି, ଯୋଉମାନେ ଏକାଠି ଯିବେ, they copulate and the eggs are released to continue the cycle once again। now this ଏଇଟା ହଉଛି egg of S cystosoma mansoni, ତା ଦେହରେ so eggs ଗୁଡିକ it is ତାକୁ ଜଣା ପଡିଥାଏ କେମିତି, by means of the type of spines that they possess। ତ pathogenicity, dermatitis ହବ, skin ଦ୍ୱାରା ମାଧ୍ୟମରେ ଯାଉଛି ମାନେ it if when it penetrates the skin, ତ ସେ ନିଶ୍ଚିତ ଭାବରେ ସେଠି ଇଚି କୁଣ୍ଡେଇ ହବ। sometimes antigenic reaction ରେ blister formation ହେଇଯିବ, ହାଡ଼ ଫୁଟି ଭଳି structure ହବ, red pimples ହବ। all these ଏରାକୁ କୁହାଯାଏ cercarial dermatitis, inflammation of the skin due to the entry of the cercaria। ବିଶେଷ ଭାବରେ lower part of the body, because they are more, when you enter the contaminated water, lower part of the body, so they find access to the lower part of the body। ତ ବଡିର ଯୋଉ limbs ରେ different region ରେ cercarial dermatitis ଦେଖାଯାଏ। ତାପରେ କାଟାୟାମାସ୍ fever ମଧ୍ୟ ଦେଖାଯାଏ due to systemic immune response। immune fever ଆସିଲା ମାନେ ଜାଣିବ immune response ଆସିଛି against the foreign antigen on the cystosomulae which enter into the body। cystosomulae ମାନେ ଏଇଟା ହଉଛି adult condition କୁ ଆସିନି cystosoma। so cystosomulae and the eggs, so it also leads to the enlargement of lymph tissue, fever ଆସିବ, splenomegaly ଆସିବ, enlargement of the spleen, hepatomegaly, enlargement of the liver, then portal hypertension, बहुत प्रकार सब pathogenicity देखा pathogenicity means what effect does it have on our body, तर body ଉପରେ ଆମର କ'ଣ ତାର ପ୍ରଭାବ ରହୁଛି। So chronic infection ମଧ୍ୟ जो रही due to the eggs entrapment within the tissue which so sometimes a eggs गुड़िक tissue रे entrap ହେଇକି ରହିଯାଏ ଛନ୍ଦି ହେଇକି ରହିଯିବ they get trapped within the tissue ଆଉ trap ହେଇକି ରହିଗଲା ପରେ it results in the granuloma formation. Okay, granuloma formation. That means cyst-like structure. So many eggs reach intestine or bladder lumen, but a large number remain stuck within the tissue and stucking ହେଇକି ରହିଗଲା ପରେ granuloma ତ formation ହବ. It will also elicit certain immune response. Our such immune response will cause the region around this egg to become fibroid. ତାକୁ କୁହାଯାଏ fibrosis ହେଇଯିବ, thickening ହେଇଯିବ। Bladder ରେ ଯଦି thickening ହବ, urinary bladder will lose the capacity for its extension। କାରଣ liver ଏ ଯୋଉ urinary bladder ଯେତିକି urine ସେ ରଖିପାରିବ, suppose you fail to void the urine, okay, you don't have a chance to go outside, ତ ସେ urinary bladder ରେ load ହେଇକି ରହିଲା କାରଣ ସେଟା extension ହେଇପାରୁଛି। If the tissue is loaded with these eggs, ତ ସେଠି fibrosis ହେଇଯିବ, fibrosis ହେଇଗଲା ପରେ it will not allow much elasticity for the extension of the bladder wall। ତାପରେ hepatic cystosomiasis, the eggs বা adult they migrate to the liver ରେ fibrosis କରିବ, ଯୋଉଠି egg trap ହେଇକି ରହିଯିବ ସେଠି fibrosis କରିବ। Fibrosis means thickening or deposition of the fibers around this surrounding in the surrounding tissue of the egg। So pulmonary cystosomiasis, hepatic cystosomiasis, pulmonary cystosomiasis ହଉଛି କ'ଣ, while they are present in the lungs ବାଟେ ଦେଇ ଯାଉଛନ୍ତି before reaching the liver, ତ ସେଠି trap ହେଇଗଲା, juvenile trap ହେଇଗଲା, ତ ସେଠି କ'ଣ ହବ, it leads to shortness of breath, ମାନେ lungs ରେ ସଦାବେଳେ ଦେଖିବ କିଛି କିଛି ଅସୁବିଧା ହେଇଗଲା ମାନେ chest pain ହବ, shortness of breath ହବ and fatigue, okay, fatigue। So detection ହଉଛି laboratory diagnosis generally କରାଯାଉଛି। laboratory diagnosis କେମିତି ମାନେ stool ଟାକୁ examine କରିବା, ଠିକ ଅଛି, stool examine କରିବା to find the presence of the eggs। ତ urine ରେ ମଧ୍ୟ there is a chance that the eggs may be present in the urine। so stool ଆଉ urine କୁ ଆମେ ପରୀକ୍ଷା କରିବା for ତାପରେ clinical manifestation, clinical symptoms ଗୁଡିକ ମଧ୍ୟ ଦେଖିବା, antibody detection ମଧ୍ୟ ଆମେ କରିପାରିବା through ELISA technique, whether antibody against cystosoma is present in the host body or in the patient body or not। Then immune evasion mechanism same like that of other drug targets madhya jo tartar emetic trivalent antimalarial monial chemical was utilized with significant adverse effect। କେତେ ଗୋଟେ drugs ଗୁଡିକ develop କରାଯାଉଛି because they have some form of adverse effect। so initially in other animals they proved very well, but in case of human being adverse effect ରହୁଛି, ତାପରେ ଆମେ ବାଧ୍ୟ ହଉଛେ ନାଇଁ this drug is not allowed to be continued। then another oxyamniquine, it was developed by Pfizer and was effective against specific Mansoni and Hematobium or Japonicum। It was used in Brazil, but କିଛି mild side effects ଦେଖାଗଲା। ତାପରେ ଗୋଟେ single dose ଇଏ କରାଗଲା। now currently praziquantel को ଇଏ କରାଯାଉଛି। praziquantel is the most appropriate drug against such parasites, ବିଶେଷ କରି cattle and domestic animals ରେ, dogs ରେ, cats ରେ praziquantel is most effective in India also। now vaccine strategy same thing, ଆମେ vaccine ତିଆରି କରିବାକୁ ହେଲେ କ'ଣ ନବା, agru koiche attenuated cercarial vaccine tamane if the vaccine is treated against the cercaria karan cercaria larva in the form of cystosomule they enter तो वैक्सीन टार्गेट करिवो तार टिश्यू को नेगो से सर्करिया लार्वा को एक्सट्रैक्ट करिकरी ताको तार टार्गेट करिबा तार टिश्यू एंटीजेनिक प्रॉपर्टी नेकी ताको एटेन्युएटेड करके वी कैन यूज एज टू डेवलप द वैक्सीन prevention and control same thing will should not allow to swim in the contaminated water clean water तापर वाटर को जदि हमें फुटे की पीवा at least 1 minute it will kill if it is boiled okay boiled means it is beyond 100 degree जदि boiling करिबा it will kill all the cercaria present ଆଉ bathing re madhya clean water stored for at least 1 to 2 days can also be used vigorous towel drying jati tame gote towel or kichi clothings use करछो accidentally joti water contaminated heichi tapare tamo vigorous towel drying after an accidental but brief water exposure may help to prevent penetration of parasites in the skin तो से ताको ड्राई करिवो से पैरासाइट गुडी को ता भितरे डेड हे जिवे बा मरी जिवे so next we'll move on to the next that is the ancylostoma duodenale it is a hookworm ancylostoma duodenale hookworm tame samaste avagata hecho in our childhood days always our parents they ask them to don't go barefoot okay don't go barefoot so barefoot in nowadays it is related to the entry of many pathogens from the soil which they inhabit in the soil now just a minute so ancylostoma duodenale it is a hookworm kahin ko agala tar body structure ta dekhibo hook shaped roichi and this they have six teeth okay six teeth both dorsal and ventral so it helps them to anchor in the wall of the intestine that is why it is called hookworm so anchoring nohele they will be eliminated from the host alimentary canal okay so causes this hookworm the disease na hochi ancylostomiasis cystomiasis ancylostomiasis or hookworm disease it is the second most common helminthic infection it is a helminth parasite roundworm parasite ascaris pore nowadays जो स्कूल चिल्ड्रेन मान को सिंगल डोज दिया जाउची माने सिंगल डोज मेडिसिन दिया जाउची अगेंस्ट दिस हेलमिंथ पैरासाइट एका दिन रे सबु पिला मान को दिया जाउची तो सेटा से इनिशिएटिव निया जाइची बिकॉज इट इज द सेकंड मोस्ट कॉमन हेलमिंथिक इन्फेक्शन नेक्स्ट टू एस्कारियासिस जो स्पीशीज हमें पढ़िबा दैट इज कॉल्ड डियोडिनेल it is a monogenetic parasite monogenetic mane gotiye animal darkar say human being hala human being so it requires only one host for the development our human hochi definitive host the human ko hi jibo though mammals like cats dogs can also be definitive host for this parasite now regarding the morphological feature um, just it is small cylindrical okay and grayish white worm covered by a cuticle kahi ki rochi for protection because it will enter another host body and syncytial epidermis syncytium means there are many nucleus syncytial epidermis anterior and ubhay male or female ro they are slightly bent in the form of a hook seitme na hela hookworm because of which they are known as hookworm they appear to be reddish brown in color when freshly passed out. Why? Because they feed on the blood only. They, the body appears reddish brown. They are passed out in the feces. ତ ଯୋଉମାନେ ବାହାରେ ଫିକାଲ ଇଏ କରନ୍ତି, ବାହାରେ ଝାଡା ଯାଆନ୍ତି, ତ ସେ ସଏଲ ନିଶ୍ଚିତ ଭାବରେ କଣ୍ଟାମିନେଟ ହେଇଥିବ। The red color is due to the ingested blood from the host intestine. Peculiarity is that they have a buccal capsule. Buccal capsule mane the cavity re which has an oral aperture. Oral aperture hoche mouth, and on the dorsal surface it has a conspicuous buccal. ତା ମାନେ ଟିକେ enlarge ହେଇକି ରହିଛି. I'll show you in my slide. Buccal capsule ରେ ଛଅଟା ଦାନ୍ତ ରହିଛି for cutting. Cutting କେତେବେଳେ? Not after entering the human body, before entering, to enter, ପ୍ରବେଶ କରିବାକୁ ହେଲେ ଯୋଉ ସ୍କିନ୍ ରିଜନରେ ସେମାନେ ତାକୁ କଣା କରିବେ, ତାପରେ ପଶିବେ। So four of these are hook shaped, while the remaining two teeth are triangular plates. The anterior end of the body is dorsally curved, which provides a grip to attach itself to the intestinal epithelium. Now here you can see the buccal capsule. So this is the true parasite. तो ଏଥିରେ ଯୋଉ କ୍ୟାପସୁଲଟା ଏଇ ରିଜନ ହଉଛି କ୍ୟାପସୁଲ, ତା ଭିତରେ these are the teeth. Here in the diagrammatic sig there are two teeth here, sorry, four teeth, two this side and two this side, and these two are dorsal tooth, which are known as the plates. Okay, triangular plates. So these teeth are prominent like the canine teeth of animals. Now next so well developed sexual dimorphism in ancylostoma duodenal में देखा जाए छे. So what is sexual dimorphism? Morphologically, if they can be differentiated into male and female worms. So here the male, the size is of the male is smaller than that of the female. So females are longer than the male. Length, um, at average, it is 8 millimeter in case of male, and in female, it is around 12.5 millimeter. Posterior end, I again, please mute. Do you like to any ask any question or mute it? Okay, thank you. Am I audible now?
Yes, sir.
Okay. So posterior end it is expanded. ଯେମିତି ତମେ ଦେଖିଲ ଆମେ ଦେଖିବା ସେଟା umbrella like manner. It contains male ରେ copulatory bursa ରହିଛି, ଆଉ female ରେ posterior end is not expanded. It is not umbrella like, and it does not have any copulatory bursa. What is copulatory bursa? The structure that help during copulation with that of the female. Now, male has a cloacal opening, female has the anus. You are all aware what is the difference between cloaca and anus. Anus is the only opening for the elimination of undigested food residue, and cloaca serves the common opening for the release of reproductive gametes as well as for the elimination of undigested food product. Then reproductive opening in case of male, it is called cloaca. Uh, in case of female, at the junction of posterior and middle third of the body. Okay, Sunanda, Sunanda, please unmute. Sunanda, you have given an. Hello, am I audible? Sunanda. Okay. Someone of you have requested for the PPT. How shall yeah, how shall I send you? Acha, Bi- Vishnu Mishra,
Sir,
I think some. Do you have any issue
हाँ
with
sir,
your phone? Acha, Sunanda Patra. Okay, we'll talk regarding the sending of this PPT. Okay. Yes, Sunanda Patra. How will I send you send this?
Sir, we have already a group in the WhatsApp.
Okay, you can add me.
Okay, sir.
You know my contact number.
No, no, sir, no, sir.
Okay, it is 94371
Yes, sir.
96322.
Okay, sir. Thank you, sir.
Okay, okay. So these are the comparative comparative account of the difference on this basis. We can it. So we come to the conclusion that they show they are highly sexually dimorphic animals. In case of male, copulatory spicules are present, which help them to transfer the male gamete to the female body, but these are not required for by the female body. Now here, this is the male and female. You can see here, the female, they have a pointed posterior end. Anus is present. In case of male, the posterior end is expanded. Both have the mouth structure. The anterior part is same, but in case of female, the posterior part and the in case of male posterior part, they are not similar to each other. Then now coming to the life cycle, okay, life cycle. So it is a very peculiar because they are monogenetic. The copulation and fertilization, they dwell in the small intestine. The copulation and fertilization between the male and female takes place in the small intestine of the human being. Now, uh, you locate the ejaculatory duct is present here, copulatory spicules is present here, and vagina is present here. So, the ejaculatory duct is present here, copulatory spicules is present here, and this is the cloaca. Now, the position of the cloacal aperture the uh, through which the sperm will be transferred, to the vaginal opening, they differ in their position. So, how will this animal copulate? And they have shown that they assume a Y-shaped structure here. So the worms, they appear like a Y-shaped structure. Okay, Y-shaped structure because of the position of the genital opening. So definitely this is the male animal and this is the female animal. So female animal kahinki ame कहले because female animal ro jo vaginal orifice ta A region re रहिछि और male animal ro jo cloaca रहिछि A region re रहिछि. So when it grabs the female in this form, the structure gives the appearance of a letter Y. So it is called Y-shaped figure. So during copulation, which occurs in the small intestine, and that too in the jejunum part of the small intestine, copulatory bursa of male coincides with the, so spicules ame kahipariba, copulatory bursa kahipariba, coincide with the vulva of the female. Vulva kahaku kahiba, opening, female opening raw jo guard type of structure roichi, that is called the vulva. Fertilize eggs, copulation heigala, eggs develop karibo. Eggs are released by the female worm in the intestine of the human and pass out along the feces. पुणी मल सहित चलीज तापर सॉइल को गला On an average, a female can lay about 10,000 to 30,000 eggs per day. गोटे दिन को 10,000 ରୁ 30,000 अंडा ଦେଇଥାଏ ଗୋଟେ female. And these freshly laid eggs are not infectious to man. Only after some time have passed, only they are infective to the human being. Further development, it occurs in the soil. Further development occurs in the soil. Con development hauchi. Now here you can see the eggs of this ancylostoma duodenale, that is hookworm. Here it is unsegmented. After the zygote is formed due to the fusion of both male and female gamete, it is a single stage. So we can say it has not gone through segmentation. Now, this is the four-celled stage where two segmentation, two cleavage division has occurred. And the larval stage that is produced is called the rhabditiform larva. Okay, this is the structure of the rhabditiform larva. This is not a diagram, rather the photograph is taken. It is the rhabditiform larva of hookworm, that is the ancylostoma duodenale. So I think it is clear that you can see the eggs, these are released from along with the fecal matter from the human body, and after remaining immediately these eggs are cannot develop if they are accidentally taken by the another human being, because it has to reach certain stages of development. Only after that it will be, it will reach the infective stage. So after entering into the human body again, it will develop. Now what will happen in the soil? What happens? So this embryo, we say embryo, even it is a four cell stage, we say it is an embryo. Embryo means it is beyond zygote stage. So embryo develops within the egg shell, that is the covering. Karana bahare, they remain outside. It has to be protected by a shell. A strong shell is present. And this embryo, the larva which is develops, it is called the rhabditiform larva, or the first juvenile stage. It is called the first juvenile stage. This rhabditiform larva, it hatches out in the soil in 2 days or 48 hours. Larva has a straight alimentary canal consisting of a mouth, buccal capsule, elongated pharynx, esophagus, everything is present in the larva. In the soil, larva to bonchiba pai kon khaibo, it feeds on the bacteria and other detritus of the soil. And this first stage larva, when it has fully grown, it molts two times. What is molting? Molting means transformation of a larval stage into another larval stage which does not resemble the adult. Adult ହେଇଗଲେ metamorphosis କୁହାଯାଏ. Molting ହଉଛି ସେ ଆଉ ଗୋଟେ larval stage କୁ, ଯେମିତି ଡ୍ରୋସୋଫିଲାରେ instar larva କୁହାଯାଏ. First instar, second ଏଥିରେ କୁହାଯିବ first juvenile stage, second juvenile stage. So first larval stage, larval stage, that is rhabditiform larva, it will molt two times. So prathama molt re second larva habo, dvitiya molt re third larval stage habo. First molt occurs on the third day and give rise to the second juvenile stage. तार कौनसी ना नहीं, larval ना नहीं, just खाली slight modification. Still it is called rhabditiform in the second juvenile stage हमें कही परिवा. Then fifth day re, third juvenile, fifth day re, the second molting hoba na, it will enter into the third juvenile stage, or it is also called filariform larva because it resembles the microfilaria of filariasis. So it is called filariform larva. It measures about very small, 500 to 600 micrometer in length, and has a sharp pointed tail. You can see here, this is the tail, this is the anterior part, and you can clearly see the alimentary canal, completely developed alimentary canal. So posterior tail is pointed, sharp pointed tail. The third stage larva, it is non-feeding. Okay, it does not feed. Rhabditiform larva, it was feeding on the bacteria. Now it does not feed. Okay, it does not feed. It is non-feeding but infective stage. Okay, it is infective stage of the parasite. That means at this stage it can, if it accidentally enters into the human body through contaminated vegetables or barefoot, barefoot, not contaminated. So it can enter and it can infect the. So it is called the infective stage of the parasite. It remains infective and alive for several weeks under favorable conditions. Uh, Favorable conditions. So direct sunlight, salt, and they do not uh, have favorable um, on this animal, and they render them they can die. So since they are alive, they are sustainable for at least for several weeks. That means the contaminated soil, it is now prone for infection for a number of days. Then infection to a new host. So new host is always the human being itself. Filariform larva, which was covered by a sheath, by a covering. Capsule no sheath. Cyst wall no. ମାନେ ସେ ଏଗରୁ ବାହାରି ଆସିଛି ରାବିଡିଟିଫର୍ମ ଷ୍ଟେଜରେ ସେ ବାହାରି ଆସିଛି ଫିଡିଂ କରୁଛି, କିନ୍ତୁ ଏଠି ଫାଇଲେରିଫର୍ମ ଲାର୍ଭାରେ ତାର ଗୋଟେ ଯେହେତୁ ଶୀଥ ରହିଛି, ମାଇକ୍ରୋଫାଇଲେରିଆରେ ବି ଗୋଟେ ଶୀଥ ରହିଛି. ଶୀଥ ମିନ୍ସ ଗୋଟେ ଥିନ୍ କଭରିଂ, ହେଲା, ଗୋଟେ ଜରି ଭଳି କଭରିଂ. ସୋ ଇଟ୍ କାଷ୍ଟ ଅଫ୍, ତାମାନେ ରିମୁଭ କରିଦିଏ ଆଣ୍ଡ ପେନେଟ୍ରେଟ୍ସ ଦି ସ୍କିନ୍. ସେଇଟା ନ ରିମୁଭ କଲେ ସେମାନେ ସ୍କିନ୍କୁ ପେନେଟ୍ରେଟ୍ କରିପାରେନି. ବିଶେଷ ଭାବରେ ଯୋଉ ଫିଟ୍ ର ଯଦି ବେୟାର ଫୁଟ୍ ରହିଲା, पूरा सोल रो तळे नुह जोटा सॉफ्ट पार्ट रही, the skin of a human host with the help of their oral spears present on the anterior end. तर anterior end रे spear माने छुरी बड़ी स्ट्रक्चर रही, ताको से यूज करके से माने पेनिट्रेट करि परिबे. बेसिकली जे माने पेनिट्रेट करंती सॉफ्ट स्किन ऑफ द फीट, हैंड रो, आउ generally through hair follicles to gain entry into the human body. Hair follicles. कॉमन साइट ऑफ देयर एंट्री होची हमरो इनर साइड ऑफ द सोल, इनर साइड स्किन बिटवीन द टोज. आंगुठी मझिरे जो स्किन रही सेटा टिके बहुत सॉफ्ट रही. बैक ऑफ द फीट रही, आउ स्किन ऑफ हैंड्स, दे आर द कॉमन. द लार्वा गेन्स एंट्री इनटू पीपल हु मूव barefoot. Yes, where good sanitary conditions do not exist. Those who do not use ସେମାନେ ପଦାକୁ ଯାଆନ୍ତି ଶୌଚାଳୟ କରିବା ପାଇଁ. Then filariform larva can also be ingested through contaminated water or food, but this is not the usual method of transmission. Yes, transplacental transmission of ancylostoma may also take place. May also take place माने establish है नहीं ये पर्यंत transplacental transmission. But the migration and later development, body को चली गला, skin को कणा करके भीतर को चली गला, सबक्यूटेनियस टिश्यू को पहुंची गला, एबे कौन करियो. लार्भा immediately ଯେତେବେଳେ skin को subcutaneous tissue रे, all tissue, you know, in between the cells there are tissue fluid. तो से tissue fluid रे रहिछि, tissue fluid are they are formed from the blood. This tissue fluid, when they are collected by specific vessels called the lymph vessel, then it is called, inside the lymph vessel, they are called lymphatic fluid or lymph fluid. So they enter into the lymphatics, into the venules. कारण वेन्यूल को गले ही से माने लंग्स पर्यंत जाई पारिबे, नेवर आर्टरी. ऑल वेन्स दे कलेक्ट द ब्लड फ्रॉम ऑल पार्ट्स ऑफ द बॉडी, तापरे से माने ताको हार्ट को पठांती, हार्ट रू लंग्स को जाए. तो लंग्स को जीवा को हेले दे हैव टू एंटर इनटू द वेन्यूल्स, वेन्स, देन कैरी टू द राइट हार्ट. राइट हार्ट काहे की कहा गला, बिकॉज़ राइट हार्ट रे, राइट साइड ऑफ द हार्ट रे द blood, unpurified blood, दूषित रक्त, unpurified blood, deoxygenated blood, they enter into the right side of the heart. तो सेठी के चली गला veins रू, from where pulmonary capillaries को lungs को गला. Larva breaks through the capillary wall. ଯେତେବେଳେ pulmonary capillary ଗଲା, capillary wall को भेद करिकरी इट विल एंटर इनटू द alveolar space alveoli भितरे से माने प्रवेश करी गले from the alveolar space the larva migrates into alveolar space माने कौन जो बट दी के एयर को जाउची एयर रो ऑक्सीजन जाउची so once they have entered into the alveolar space now they can enter into the bronchi. Okay, so they migrate into the bronchi माने wind pipe ହେଲା प्राइमरी ब्रोंकाई सेकेंडरी रही रेस्पिरेटरी ब्रोंकियल्स ये सब पढ़ी सो दे एंटर इनटू द ब्रोंकिया एंड ब्रोंकिया परे कोन आसीबो ट्राचिया ट्राचिया माने बेकरे जोटा रही विंड पाइप इट डिवाइड्स इनटू टू ब्रोंकस सो ट्राचिया को आसीगला ट्राचिया आसीगला ट्राचिया कोठी खोलीची तंटी पाखरे खोलीची नियर द इट रीचेस द लैरिंग्स ओपन नियर द फेरिंग्स आउ फेरिंग्स रीजन रे लैरिंग्स को जेतबले आमे फूड आमे स्वालो करूचे the windpipe is closed by epiglottis. Okay, so epiglottis ताको बंद करे तो विंड पाइप रो निकटतम हो जाइछि एपिग्लॉटिस सो विंड पाइप रो लैरिंग्स रो विंड पाइप रो प्रथम ओपनिंग टाको लैरिंग्स कहते तो लैरिंग्स पाखरे epiglottis रही they move into the epiglottis wriggle over the epiglottis and back of the pharynx where सेतबेले कोन हबो सलसल हबो छोट पिला माने से माने कास हबो okay they cough so when they cough the children um, the children generally swallow it the children generally swallow it से कासिकिर पका दिने राति बेले हबो कासिकिर से मन डुकी दियंती तो चाली गला कोठी की it reaches the intestine so when the third instar larva reaches the esophagus it molts for the third time to form fourth instar larva ए पर्यंत एंट्री कला थर्ड इंस्टार लार्वा रे फाइलरी फॉर्म लार्वा रे ए प्रकार सबु जगह बुली किरी फाइनली आसी गला पाटी पाखरे तंटी पाखरे आसी गला तंटी पाखरे टिके सलसल हे गला कास हेला कास हेकरी से चली गला into the body into the alimentary canal through the esophagus तापरे it reaches the esophagus it molts for the third time तृतीय थर आउ fourth instar larva formation हे गला and this fourth instar larva moves into the small intestine where it finally settles down and undergoes final molt fourth molt रे fifth larva formation of third molt रे fourth instar larva fourth molt रे final molt रे it will transform into an adult and here with the help of its structures it will remain attached to the wall in about 3 to 4 weeks time they become sexually mature to repeat the cycle they repeat the cycle so here the representation of the life cycle you can see here these male and female inside the body the eggs are released into the soil प्रथम राबडीटी फॉर्म लार्वा हेगला तापरे दीटा मोल्ट हेकरी फाइलरी फॉर्म लार्वा हेगला and this filiform larva they enter through the soft tissue of the toe and it has a blood vessel lymphatic system enters blood and lymphatic system to reach the right side of the heart ro lungs को गला ए lungs रे alveoli ru bronchus बाट देकरी trachea बाट देकरी larynx को आसी गला larynx is nearer to the pharynx सेठी पहुंची गला परे कास हबो कास हेकरी पुणी से माइक्रोफाइलेरिया फाइलेरियासिस larvae filiform larvae are swallowed so generally unhygienic activities by barefoot movement छोटो पिलांकर देखा जाए so mainly these small children से माने कासिगिरी सेटा ढोकी दियंती they are infected to this disease so pathogenicity रे एठी देखा जायछि जेतल cutaneous larva migrans caused by ancylostoma duodenal से ए जो eruption ही जायछि देखिबो in the skin and here larva are present जो in the lung tissue ठीक अछि lung tissue रे से माने alveoli रे पहुंची जायछथि देखो जो larval of the ancylostoma duodenal, how they look like. Now, regarding the pathogenicity, I have told you बहुत blood loss हुए जो ancylostoma duodenal रे, because say blood गुड़िक they are lost at the rate 0.2 ml per blood per day. uh, and the adult it sucks about 0.2 ml of blood per day. तो ଯେତେଟା adult ରହିଥିବେ ସେତିକି ପରିମାଣରେ acute in symptoms include nausea, vomiting, chills and fever, କମ୍ପ ଲାଗିବ, ଜ୍ୱର ହବ, କାଶ ହବ, abdominal pain ହବ, ulcer ହବ, intestinal cramps ହବ, ପେଟ କାଟିବା, loss of hunger, blood in the stool, ଯଦି ଯୋଉଠି କଣା କରିଥିବେ ବ୍ଲଡ୍ ସେଠି ଯଦି ଅତ୍ୟଧିକ ସେଇ ଜାଗାରେ ବହୁତ ଗୁଡେ ଆନ୍ସାଇଲୋଷ୍ଟୋମା ସେମାନେ ଆଟାକ୍ କରିଥିବେ ତ ଅଧିକ ବ୍ଲିଡିଂ ହବ ଆଣ୍ଡ ଦିସ୍ ବ୍ଲିଡିଂ ବ୍ଲଡ୍ ଇନ୍ ଦି ଷ୍ଟୁଲ ସେ କଣ୍ଡିସନରେ ଦେଖାଯାଏ। କ୍ରୋନିକ୍ ସିମ୍ପଟମ୍ସରେ ଆନିମିଆ ହବ, tachycardia means heart will beat faster, then dyspnea, shortness of breath, କାରଣ ଯେତେବେଳେ ଲଙ୍ଗସକୁ ଯିବେ ଲଙ୍ଗସକୁ ଇଏ କରିବେ ତ ବ୍ରେଥଲେସନେସ୍ ଲାଗେ, ତାକୁ କୁହାଯାଏ dyspnea. Excessive blood loss caused by prolonged worm infection leads to hypochromic microcytic anemia. ତା ମାନେ ହଉଛି ଆରବିସି ରେ ଯେତିକି ହେମୋଗ୍ଲୋବିନ୍ ରହିବା କଥା, ଗୋଟିଏ ଆରବିସି ରେ ଯେତିକି ପରିମାଣର ହେମୋଗ୍ଲୋବିନ୍ ରହିବା କଥା ସେଟା କମିଯାଏ, so it is called hypo. And other effect, malnutrition, malabsorption of the food. Okay, so diagnosis haba, examine kariba, stool examine kariba, to stool re if you find these rhabditiform sorry eggs, generally eggs uh, are present, tale jana padibo stool. So if eggs are not present ଆଉ କିନ୍ତୁ ସେ ପ୍ରକାର symptoms ରହିଛି continuously, they go for the x-ray. So chest x-ray ରେ ଆମର lungs ରେ ତାର position ଆମେ ଜାଣିପାରିବା, which is corroborated with the clinical symptoms. ତାପରେ mainly severe anemia ମାନେ ଏଇ 11-12 ନୁହଁ, ବହୁତ କମିଯିବ, 7-8 କମିଯିବ severe anemia. And high levels of eosinophilia, high levels of DC blood ର ଯେତେବେଳେ DC count କରାଯିବ, eosinophil count ଯେତିକି ରହିବା କଥା, actually eosinophil should be around 4%, ତ ସେଟା ବଢିଯିବ ପୁରା 12, 14, 16 ଏମିତି। then treatment ହଉଛି generally mebendazole. So all of you must be aware of it. A dose of 100 milligram is given twice daily for 3 consecutive days. So it is the best drug for against this hook. And in addition, there are other parental pamoate are also given. Immune response nishchita bhabe rohichi because they have antigen on their body, because any foreign body ama body re pasile immune ye rahibo nishchita bhabare, and they initiate the humoral response or the cellular response depending upon the production of T cell or B cell. The cellular response re T cell, B cell seଗୁଡ଼ିକ produce ହୁଏ, ଆଉ humoral response re generally the antibodies are produced against the organism, and these antibodies are produced by the plasma cells. Next, vaccine, we move on to the Trichinella spiralis. So this animal, it is a peculiar animal. The name itself implies that it is a hair-like animal which is coiled, okay, which is spirally coiled. So trichos means hair, and ella spiralis is a Greek term, means spirally coiled. So ଏଥିରେ 8ଟା species ରହିଛି। So the common ହଉଛି spiralis, ଯୋଉଟା ଆମେ mainly study କରାଯାଇଛି life cycle ସବୁ ଗୁଡ଼ିକ। host ହଉଛି definitely human being, ନହେଲେ ଆମେ କାହିଁ ପଢିଥାନ୍ତେ, but domesticated domesticated animals are also found. These include the birds, mice, pigs, cats, dogs. Pigs serve as the primary host of Trichinella spiralis and as the reservoir host for humans. So primary host hochi the pigs, a reservoir host hochi human. What is a reservoir host? Reservoir host hochi those organisms which harbor the host for its transmission to other animals. Taku kuhajaye reservoir host. Our habitat hochi they always remain in the mucosa, okay, mucosa of the small basically small intestine of definitive host. The larvae insist themselves in the striated muscles re semane rohanti. They live in the muscles, okay, well protected in the muscles. তো অ্যাকচুয়ালি প্রথম থর যেতবে আবিষ্কার করা গলা দি স্যার রিচার্ড ওয়েন, so when he was a physician, autopsy କରୁଥିଲେ ଗୋଟେ ଇଟାଲିଆନ ପେସେଣ୍ଟ ଯାହାର ଟିବି ରେ ମରି ଯାଇଥିଲା ସେ, ତ ଯେତେବେଳେ ଅଟୋପସି କରାଗଲା and when they studied the diaphragm, they found this Trichinella spiralis in a spiral manner present in the diaphragm. Diaphragm জানছো তো, it is a muscles present in the lower abdomen which helps for inspiration and expiration along with the intercostal muscles. তো diaphragm রে এই প্যারাসাইটটি রহিଥିଲା ট্রিচিনেলা স্পাইরালিস। তো সেইটু জনা পড়লা যে দিস animal or this parasite is a human parasite. That does not mean that it is responsible for the tuberculosis. সেটা পে নুহ, কিন্তু tuberculosis রে সে পেশেন্ট ইটালিয়ান পেশেন্ট মরি যাইଥିଲା, তাকে অটোপসি কলে স্যার রিচার্ড ওয়েন, এবং অটোপসি কলা পরে সেটা দেখিলে যে তার ডায়াফ্রাম মসল রে সে ইয়ে রয়েছে। এনিওয়ে, humans can get infection orally by eating raw meat, okay, or generally raw meat तो ये करते और poorly or undercooked meat, माने संपूर्ण भावरे कुकिंग है नहीं, भीतर से पैरासाइट रो जो स्टेज रही छी सेटा मरी नहीं, सेटा जीवित रही छी, and it is capable of infection. So poorly or undercooked meat of infected animals, जो animals माने infected है थी। Now here you can see the morphological feature of Trichinella. So as the name implies, se pura spiral ହେଇକି ରହିଛି, okay, spiral ହେଇକି ରହିଛି. So it is not the diagrammatic, it is the true picture. So it is a tissue nematode. It is the causative agent trichinosis, Trichinella ରୁ trichinosis ହବ, filaria ରୁ filariasis, ascaris ରୁ ascariasis. So trichinosis is the disease caused by Trichinella spiralis. It is spirally coiled, appearance of the larva in the muscles. এই অবস্থাରେ ସେ larva গুড়িক দেখাଯାଏ muscle ভিতরে। common name হচ্ছে Trichina worm. Now morphology, এটি দুইটা যাক adult ଆଉ adult worm, both male and female এটি দেখা যাইছি। adult worm, it is small white worm, just visible to naked eye, ଗୋଟେ speck ଭଳି ଦେଖାଯିବ, ସମ୍ପୂର୍ଣ୍ଣ ଦେଖାଯିବନି, just visible to the naked eye. It is one of the smallest nematodes infecting humans. The anterior half of the body is thin and pointed, well adapted for burrowing into the mucosal epithelium. କାରଣ mucosal epithelium କୁ ଅତିକ୍ରମ ନ କଲେ ସେମାନେ muscles ଭିତରେ ପହଞ୍ଚି ପାରିବେ ନାହିଁ। So this is the female animal and this is the male animal. So nematode रे देखिबो जो dimorphism रहिछि, एथरे भी से प्रकार sexual dimorphism देखा जाए। Females have a longer body than that of the male. Female रे anus रहिछि, male रे cloacal aperture रहिछि. These are the basic here. Then here the more distinct diagram is given here. This is the male and this is the female. You can see how elongated it is, and these are the embryos present in the anterior. So this is the anterior part where you find the embryo which have not developed, and these are the larval stages which have developed, and they are viviparous in nature. एमाने जन्म दियंती छुआ माने एटी एक्चुअली कॉपुलेशन होची एग डेवलपमेंट बॉडी भीतर है कि बॉडी भीतर हैचिंग है कि दी larva comes out through the opening, that is the vulval opening to the outside. So NBL means newborn larva. Okay, newborn larva. এটা হচ্ছে ম্যাচিওর লার্ভা। এমএল মানে হচ্ছে ম্যাচিওর লার্ভা। ম্যাচিওর লার্ভা এটা ସେମାନେ ଫ୍ରିରେ କେବେ ରୁହନ୍ତି ନାହିଁ। ଦେ always remain within the muscles in a twisted manner. Okay, so I think you are well aware of this structure. Both male and female, they show well developed sexual dimorphism. So here the comparison is made here between the male and the female. The size ଦେଖିବ almost double size ରହିଛି female than that of the male. So it is जो star mark রয়েছে actually এটা হচ্ছে the diameter. Okay, this is the length, this is the diameter. So female diameter is greater than that of the male কারণ it is overladen with the developing egg and the embryo. So half length রয়েছে male রে clasper রয়েছে, clasper helps in the transference of the sperm into the female body. The female worm is viviparous, so it is the only worm that is viviparous. It eggs develop within the body of the female, and the larvae hatches out and comes out through the vulval opening. Male worm dies soon after fertilizing the female. Female dies after 4 weeks to 4 months, the time required for discharging the larva. Now, tell me, male dies after fertilizing the female, and after a single one time fertilization the male dies. সেই ଫର୍ଟିଲାଇଜେସନରେ ଯେତେ ଏଗ୍ ରହିଛି ସମ୍ପୂର୍ଣ୍ଣ ଭାବରେ ସବୁ ଏଗ୍ ଗୁଡ଼ିକ ହ୍ୟାଚିଂ ହେଇ ନ ହେଇ ସାରିଲା ପର୍ଯ୍ୟନ୍ତ ସିଏ ବଞ୍ଚିଥିବ। ହେଲେ ସବୁ ଏଗ୍ ହ୍ୟାଚିଂ ହେଇଗଲା, ଲାର୍ଭାଲ ଗୁଡ଼ିକ ଜନ୍ମ ନେଇଗଲେ, ତାପରେ ଫିମେଲ ମରିଯିବ। There is another animal where the male dies after copulation, that is the honeybee. Okay, that is the honeybee. Now the larva again, this is the stained within the muscles. Muscle bhitere kemti larva ta twisted heiki roichi, and this is the embryos discharged by the female measure only and are spiral in shape, hence the name spiralis. They are discharged by the female worm in the host intestine, from where they are carried by the circulating blood to the striated muscles where they encysted themselves. Encysted ମାନେ ନିଜକୁ କଭର କରିକି ରଖିଦେବେ, ଗୋଟେ ହାର୍ଟ ଟାଇପର କଭରିଂ କରିକି କନସିଲ୍ କରିକି ରଖିଦେବେ in the muscles. So that is why the name Trichinella spiralis. ତାପରେ ଦେଖ ଏଇ ଯୋଉ ସିଷ୍ଟ କଣ୍ଡିସନରେ ଯୋଉ ଏନସିଷ୍ଟ ଆମେ କହୁଛେ, the true cyst ଏଠି ଦେଖାଯାଇଛି। ଦେଖ, this is the animal coiled ହେଇକି ରହିଛି, this is the cyst wall. So it is the tissue reaction around the encapsulated larva. जो टिश्यू रिएक्शन माने सराउंडिंग टिश्यू रिएक्ट करके ता चारिपटे गोटे सिस्ट कवरिंग, कारण से सब बेले पूरा कंप्लीटली परमानेंटली से मसल भितर रही गला। इट डेवलप्स प्रिफरेंशियली इन एक्टिव मसल्स। कोठी रहिबो एक्टिव मसल जो माने नियमित भाबरे काम करंती, और नियमित भाबरे काम करे हमरो डायफ्राम, कारण diaphragm always in an active state because for it helps in during respiration. Jaw muscles भी हमें सदा बेले कथा कहला बेले खाइला बेले every time jaw muscles, bicep muscle माने hand row muscle, neck row muscle, lower back, which are relatively poor in glycogen and hypoxic environment रे से माने रुहंती, more abundant near the site if attachment of muscle to tendon, muscles to tendon. Okay, muscles to जो site रे if attachment of muscle to tendons and bones and lie longitudinally in muscle fiber. Longitudinal माने parallel to the muscle fiber. Now तार life cycle, human being रे life cycle गोटे host दरकार, as I have already told you. Another host is required only for the perpetuation. Perpetuation माने कौन? Suppose मनुष्य देह को चली आसिला, मनुष्यर muscles रे रहिला, मनुष्यर फ्लेश तो के खाइबे नहीं ना। तो जनरली परपेचुएशन माने बाय सम मीन्स अन्य एनिमल रे जदी रहिला से अन्य एनिमल को केमती गला ना, जो एनिमल को गला इट मस्ट बी ए फ्लेश ईटिंग एनिमल, आउ ताठारु जो एनिमल को जीव सेटा भी फ्लेश ईटिंग। सो आमे कहिबा इट मूव्स फ्रॉम ए फ्लेश ईटिंग एनिमल टू ए फ्लेश ईटिंग एनिमल। तो ह्यूमन बीइंग को सपोज ह्यूमन बीइंग इफ इट इज टोटली, if one is totally herbivore in diet, माने नियमित भाବରେ से केबे हेले आइष खाएनी, निरामिष खाए मूलरू, तो निश्चित भाव there is no chance for this trichinosis. So if at all ତ ମଣିଷ ଦେହରେ ସେଟା ହଉଛି ବ୍ଲାଇଣ୍ଡ ଏଣ୍ଡକୁ ଆସିବ, କାରଣ ଦେର୍ ଇଜ ନୋ ଅଦର ୱେ। ସେ ପରପେଚୁଏଟ ହେଇପାରିବନି, ସେ ସେଇଠି ରହିବେ। ସେଠି ରହିଲେ ବି ଟିଲ୍ ହ୍ୟୁମାନ୍ ଡାଇ ହେଇଯିବ, ସେ ମରିଯିବ। There is no intermediate host. However, the parasite entering human cannot complete its life cycle. ସେଥିପାଇଁ କୁହାଗଲା it needs another animal for continuance. Pig serves as a reservoir host for man. Man ର pig ହଉଛି reservoir host. So pig serves as a reservoir host of man because man has the habit of taking undercooked pork. The intestinal phase, human acquire the infection by eating undercooked. ତାପରେ undercooked ସେ ଯୋଉ ମସଲ ଭିତରେ ରହିଲା ନା, ଇଏ ହେଇକିରି ସେ ମସଲ ଭିତରେ ରହିବା ଅବସ୍ଥାରେ it enters into the stomach. Digestive juices of the stomach ତାର collagen capsule ବା ତାର ଯୋଉ cyst covering ଟାକୁ digest କରିଦେବ, and digest କରିସାରିଲା ପରେ the larvae are released from the cyst and the process is called excitation. ରିଲିଜ ହେଇଗଲା ପରେ ସ୍ମଲ୍ ଡିଓଡିନମକୁ ଜିଓଜିନମକୁ within 24 hours to 30 hours they mold 4 times. 4 ଥର ଚେଞ୍ଜ ହବ ଏଇ ancylostoma ଭଳି to become adult worm. ତାପରେ they bore their way into the columnar epithelial cells of the mucosa and attain sexual maturity. attain କରିଯିବେ male and female. fertilization occurs after about 24 hours of attaining sexual maturity. ଆସିଗଲା ମାନେ fertilization ହବ ଆଉ କଣ, after which male ମରିଯିବେ। ସେମିତି କହିଥିଲି ଯୋଉ ହନିବିରେ ମଧ୍ୟ ମେଲ୍ ମରିଯାଏ ଆଫ୍ଟର କପୁଲେସନ୍। ଏମ୍ବ୍ରିଓଜେନେସିସ୍ ଷ୍ଟାର୍ଟ କରିଯିବ, ଲାଷ୍ଟ ଫର୍ ଆବାଉଟ୍ 4 to 5 days। ଆଣ୍ଟିରିୟର ଏଣ୍ଡରେ ଏମ୍ବ୍ରିଓ ରହିଥିବ, ପୋଷ୍ଟିରିୟର ଏଣ୍ଡରେ ସେ ଏଗ୍ରୁ ଛୁଆ ବାହାରିକିରି ସେମାନେ ଜନ୍ମ ନେଇଯିବେ। ସୋ ଦେ ଆର୍ viviparous. A single fertilized female lays about 1500 larvae in 1 to 3 months. Muscle phase of the life cycle, newly emerged larva possess stylate in their oral cavity. Stylate माने गोटे spear like structure जहाँ द्वारा से माने lymphatic vessel को ଯାଇକିରି mesenteric vein तापरे musculoskeletal cells को पहुंच जिबे, सेठारे muscle fiber ର parallelly ସେମାନେ arrange ହେଇକି ରହିବେ। The presence of the larva, it causes leads to disappearance of the myofilament, degeneration of mitochondria and enlargement of the nuclei to form a complex capsule around the larva. କିଛି ଚେଞ୍ଜେस ଆଣିଥାଏ ସରାଉଣ୍ଡିଂ ଟିସ୍ୟୁରେ, ଯାହାଦ୍ୱାରା କି ସେ ଲାର୍ଭାଟି ଅଟୋମେଟିକାଲି ସେ ଏନସିଷ୍ଟ ହେଇଯାଏ, ତା ଚାରିପଟେ କ୍ୟାପସୁଲ ଫର୍ମେସନ ହେଲା। ଏଇ ଏନସିଷ୍ଟେଡ ଲାର୍ଭା ବା କ୍ୟାପସୁଲେଟେଡ ଲାର୍ଭାକୁ ଏ ଅବସ୍ଥାରେ ତାକୁ କୁହାଯାଏ nurse cell. In about 3 to 4 weeks, the larvae are fully developed and a double-walled capsule is secreted around them. Capsule formation is completed in about 3 months. Encysted larva se parallelly muscle fiber ରହିବ, ସେ non-pathogenic for the natural host except for human. Non-pathogenic for natural host except for human. Human ରେ pathogenic ରହିଛି, pathogenic ମାନେ it will bring about certain symptoms. The most preferred site of Trichinella larvae in human are the striated muscle of the diaphragm, intercostal muscle, eye muscle, जोटा muscle गुड़ी को सबले व्यवहार हुए और जोटी ग्लाइकोजेन अभाव रहुची और जोटी ऑक्सीजन अभाव रहुची। tongue muscles सबले व्यवहार होची। the muscle phase marks the dead end, आउ से human being रे जति रहिला आउ कुआडे जाई परिबो नाही। Where is the cursor? Just a minute. Life cycle, the pig is the favorable host, alternate host is man, and man is the dead end, and infective form hochi encysted larva in the muscles. So the flow chart is given here. Meat is taken, undercooked meat without adequate cooking. Cysts are digested by the gastric juice, and these are viable larvae are released. Then they penetrate the mucosal epithelium of the intestine, mold 4 times, and they become sexually mature. The male dies. So when they sexually mature, they copulate. Male dies after fertilization, and larva continue to discharge during the lifespan, that is the 4 weeks to 4 months. Larva enter the intestinal lymphatics ହେଲେ ତାପରେ ସେ ତାର ମସଲ ପର୍ଯ୍ୟନ୍ତ ପଳେଇବ, ମସଲ ଭିତରେ ସେ ଏନସିଷ୍ଟେଡ ହେଇକି ରହିବ। ଇନ୍ ମ୍ୟାନ୍ ଦି ଲାଇଫ୍ ଏଣ୍ଡସ୍ ହିୟର, କାରଣ ମ୍ୟାନ୍ ରୁ ଆଉ ଇଏ ଯିବ ନାହିଁ। କ'ଣ ମ୍ୟାନ୍ ଇଜ ନଟ୍ ମାନେ ଆଉ କିଏ ଭକ୍ଷଣ କରିବେ ନା ଅନଲେସ୍ ଆକ୍ସିଡେଣ୍ଟାଲି। ସୋ ହିୟର ଚିତ୍ରଟି ଦେଇଛି, ଦେଖ କେମିତି ଷ୍ଟୋମାକ୍ ଭିତରେ ଏନସିଷ୍ଟେଡ ହେଇକି ଡିଜଲ୍ଭ ହେଇଗଲା, ତାପରେ ତାର ଲାର୍ଭା ଗୁଡିକ ବାହାରି ଆସିକିରି, ଏଇଟା ହଉଛି ଇଣ୍ଟେଷ୍ଟିନାଲ ମ୍ୟୁକୋସା। ସୋ ଦେ କ୍ରସ୍ ଦି ଇଣ୍ଟେଷ୍ଟିନାଲ ମ୍ୟୁକୋସା, କମ୍ ଟୁ ଦି ଭେନ୍ୟୁଲ୍ସ, ବ୍ଲଡ୍ ପାଖକୁ ପଳେଇ ଆସିଲେ, ତାପରେ ଅଲ୍ଟିମେଟଲି ଦେ ଫାଇଣ୍ଡ देयर वे। ଏଟା ହଉଛି ଏନବିଏଲ ମାନେ ନ୍ୟୁ ବର୍ନ ଲାର୍ଭା, ଦେ ଫାଇଣ୍ଡ देयर वे in the muscles। ଦେଖିବ ମସଲ ରେ এটা হচ্ছে muscle fiber, so they are present parallelly, and they are oriented parallelly to the muscle fiber। ଆଉ ଟିକେ ଏକ୍ଜାକ୍ଟଲି ଦେଖାଯାଇଛି ଯୋଉ 1 ନମ୍ବରରେ ମଣିଷ ଦେହକୁ ଯିବ by eating improperly cooked pork, ତାପରେ ମଣିଷ ଦେହରେ release of infective larva of 2 ନମ୍ବରରେ ଲାର୍ଭା ଗୁଡିକ ବାହାରି ଆସିଲା, which is taken from the intestine। the photo, the migration of the to the small intestine, तापर से small intestine रे एठी पहुंची गला, एटा enlargement करा जायछि। small intestine में पहुंची गला परे mature हे जिवो, mature हेला परे the again copulate in the body, and after copulation the newborn larvae NBL are released, and they move into the suitable muscles where they become encysted। Now, pathogenesis of the clinical features pathology. The first stage begins with ingestion of raw pork containing larva and ends with invading the intestine and developing into adult. This stage begins when new infective larva released. So, stage of muscle invasion and stage of, so this is the pathology. Even if they enter into the muscles, thanks they never enter the myocardial muscles। if they enter into the myocardial muscles, सेठी भी जो ये ହେଇଯିବ କ'ଣ ନର୍ଭ ସେଲ୍ ଫର୍ମେସନ୍ ହେଇଯିବ, ତା ଦେହରେ ସିଷ୍ଟ ଏନସିଷ୍ଟେଡ କଣ୍ଡିସନରେ ସେ ମସଲ୍ସ କାର୍ଡିଆକ୍ ମସଲ୍ସ କାମ କରିବ ନାହିଁ। Now clinical features, intestinal invasion ରେ malaise ମାନେ ପଳିଯିବ ଦେହରେ, ରେଡ୍ କଲର vomiting ଭଳି ଲାଗିବ, ବାନ୍ତି ହବ, diarrhea, abdominal cramps, and it is onset within 2 to 3 hours of ingestion of infective food which is infected with trichinella। Then second stage of muscle invasion, fever, periorbital edema। periorbital edema माने आखिर जो eyeball रहिछि सेथिरे if the water fluid accumulates, the चारिपटे edematous eye देखा जिवो आखि। weakness of affected muscle, जो muscles रे से माने रहिथिबे, so suppose diaphragm रे बहुत अत्यधिक परिमाण रे accumulate करि गले diaphragm ठीक भाबरे contraction करिवो नै, contraction न कले respiration भलो भाबरे हबो नाही। So myocarditis, so generally it does not move into the, if it enters into the heart muscle, it leads to myocarditis if heart muscle is involved। Encephalitis means if it involves the brain, enlargement of the brain। Eosinophilia is a constant feature where the number of eosinophil cell increases। Diagnosis, muscle biopsy करिबा। Okay, muscle biopsy, stool examination, xenodiagnosis mean any other foreign gene is present। foreign gene eti mane kon, xenodiagnosis you have taken some other अन्य प्रजातिर animal को नेइछ with the tissue ताको examine करि परिबा। the direct indirect previous history of your eating habit, blood examination ରୁ ଜାଣି परिबा, plasma examination ରୁ serology, then radiological examination, radiology means CAT scan ହେଲା, x-ray ହେଲା, ସେଥିରେ muscles ରେ ଏମାନଙ୍କ ଉପସ୍ଥିତି ଦେଖା ଯିବ। ତାପରେ PCR ରେ जो एंटीजेन बा तांकर जो डीएनए गुडी को एम्प्लीफाई करिके तक हम ଇଏ କରିପାରିବା। Then all these are directly ତାର ଇଏ କରାଯାଇଛି। History of eating raw or uncooked, blood examination is. Then treatment, radiological calcified cyst can be seen. Treatment mild cases re supportive treatments like bed rest, nobo, analgesic. Analgesic mane if you have temperature, go for paracetamol and antipyretics. Moderate cases re you can give albendazole, provided it is it is established that one is, I mean, infested with this Trichinella. Mebendazole can be given. Uh, Severe cases add glucocorticoids. I mean, you give steroid. Okay, in severe cases, steroids may be given like prednisolone and 2-albendazole or mebendazole. So, along with albendazole or mebendazole, prednisolone is a most rather you can say steroid, very safe steroid, prednisolone. It can be given around I think it is severe case is 16 milligram in single dose for a number of days. Prophylaxis prevention, don't eat pork. Um, Stop the practice of feeding pigs with raw garbage. Extermination of rats from pig farms spread. Smoking, salting, and or drying the meat does not destroy the infective stage. ମନେ ରଖିବ ଯୋଉ ସଲ୍ଟିଂ କରିଦଉଛେ ସେ ଇନଫେକ୍ଟିଭ ସ୍ଟେଜ କୁ ମାରି ଦିଏ ନାହିଁ। Prolonged freezing decontaminates the prolonged freezing mane freezing for at least 1 day, I think, in -5 degrees centigrade it will decontaminate the meat. Now next is the most important, that is Entamoeba histolytica. You are all aware of this disease or this phenomenon, and one may acquire this disease very frequently of your feeding habit. You go for taking pani puri or something like that, because they use this contaminated. So mostly when you think that how I have contracted this dysentery, you remember that whether you have shared something or you have gone for any other roadside food. So entamoeba is an anaerobic parasite. Humans re entamoeba. Since it dwells in the intestine, particularly in the colon region, gastrointestinal symptoms are prominent. It causes amoebiasis, acute and chronic amoebic dysentery, enlargement of these liver, spleen, brain if it enters. Lesions means lesions, abscess means a structure which encloses some fluid. ତାକୁ abscess କୁହାଯାଏ। ପୂଜ ଭଳି ପୂଜ accumulate କରିଗଲା ଭଳିଆ ସେଟା ତାକୁ abscess କୁହାଯାଏ। It is cosmopolitan, globally ଦେଖାଯାଏ। Discovered by Lambie during an autopsy. ପୁଣି ସେ autopsy of the colon of a child who had died of diarrhea. ତ ସେ ନିଶ୍ଚିତ ଭାବରେ ଏଣ୍ଟାମିବା ହିଷ୍ଟୋଲିଟିକା ଦ୍ୱାରା ଇନଫେକ୍ସନ, ତାର କୋଲନ ରୁ ମଧ୍ୟ ଏଇଟା ମିଳିଲା। ସେଇ ଦିନ ଠାରୁ ଜଣା ପଡିଲା ଯେ ଦିସ୍ ଏଣ୍ଟାମିବା ହିଷ୍ଟୋଲିଟିକା ଇଟ୍ ଇଜ ଏ ଭେରି ସାଂଘାତିକ disease, very deadly disease. So it causes amoebiasis. Around 10% of the world population is infected. 10%. ଅଧିକାଂଶ କ୍ଷେତ୍ରରେ ତମର ଆମର ରହିପାରିଥାଏ, but symptoms around 90% they are asymptomatic. They don't show any symptoms. Deaths have been reported globally only in about 10% of the infected people, because all of now the WHO, they have rendered supply of medicines, and the protocol is maintained. Entamoeba histolytica, it is also a monogenetic parasite. So two monogenic parasite, common host are human being, apes and monkeys. It exists in two forms, minuta and virulent form. So as the name implies, minuta is non-virulent. That means it is non-pathogenic. It cannot invade. It is non-invasive form. A virulent form is the pathogenic form, and that lives in the epithelial cells of the mucosa and submucosa of the large intestine. That particularly large intestine, cecum, colon, and sigmoid rectal region. Sigmoid colon, cecum. cecum region hochi where the ileum joins with the colon. That part is called the cecum. From the cecum arises the appendix. तो सेठी रहंती एमने सेठी रहंती बेले बेले appendix को एमने affect कर दियंती. They invade the intestinal mucosa and spread to other organs like liver, lungs, heart through the bloodstream. Now, here you can see the structure of a large intestine. The large intestine, this part is called the colon, okay, cecum. Okay, this is the cecum. Yeah, ascending colon, transverse colon, descending colon. This is called sigmoid colon also. Okay, this sigmoid colon, A region and this region, these parasite cysts are present here. Next, the morphological features, if we study about entamoeba histolytica, generally three features, structures are seen. That is the trophozoite stage, cystic stage, and the pre-cystic stage. So, cystic stage is the final stage beyond pre-cystic. Pre-cystic is transient stage between trophozoite and cystic. So means a trophozoite will transform itself into a cystic form through pre-cystic. So trophozoite is the invasive. It is the invasive form of the parasite. It is the invasive form. That means it is the active form. It is the feeding form, invasive form of the parasite. And definitely gotiye nucleus roite by which you can identify the entamoeba from other such protozoans. It has a prominent nucleus. uh, Beside nucleus, endoplasm also contains the ingested red blood cell. So it appears red in color. Red blood cell, food vacuoles রয়েছে, granules, tissue debris রয়েছে. So very important, and one of the most important thing is that electron dense granules which have proteolytic activity. Proteolytic activity means they can digest the protein. So with proteolytic activity, these granules have been found on the surface of the plasma membrane, by which it is able to invade the lining of the intestine or the colon. These trophozoites go on dividing every 8 hours by binary fission, every atagota. But it will not continue. And after some time, some of them go on dividing, others stop dividing, and they transform themselves into the cystic stage through the pre-cystic stage. Here you can see the trophozoite stage. This is the normal stained stool niajaichi entamoeba, and this is the diagrammatic where you can find a pseudopodium. It is the trophozoite. These are the food vacuoles which are ingested, which contains blood cells. Yeah. And this is the nucleus of the animal. Now, some of the trophozoites which are active stage, they transform into pre-cystic form, that is called the minuta form. They pre-cyst is the intermediate, okay, just it is in a transient stage. It is a preparatory stage where it will remove all the food vacuoles and become prepared to undergo encystment. So trophozoite undergo encystment in the gut lumen. Prior to encystment, trophozoite exudes, it removes food vacuoles, becomes round, ovoid. This is called the pre-cystic stage of the parasite. It is smaller in size. Then nucleus, there is no change in the nucleus except it becomes larger. There is no discrimination of the, both are surrounded by ectoplasm, endoplasm. A covering called pellicle is present, a small pseudopodia is present, but they don't have any food vacuoles in them. They have removed the food vacuoles. They have extruded the food vacuoles, and due to which they have become rounded in structure. Now the cystic wall, that means complete cyst has been formed. Um, Highly refractile, hard covering, because they have to remain outside the human body in the soil. That is why in a preparation, they develop, the trophozoite develop a cyst. It makes the cyst resistant to the gastric content of the stomach, adverse environmental condition, and to the chlorine concentration found in the water. So it is resistance to chlorine also. So cysts of the amoeba histolytica are found in the lumen of the large intestine. ସେଇଠି formation ହୁଏ, ସେଇଠି ଦେଖାଯାଏ। So if you want to test the go for stool testing, so the watery stool during amoebic dysentery, you will see highly refractive shining cystic which are oval in shape ସେଥିରେ ସେ ଫିଲ୍ମରେ ଦେଖାଯିବ। This cyst, they initially they are uninucleate, they are which is mature immature, then become binucleate, dita nucleus habo sei gotiye cyst re, ତାପରେ ଚାରିଟା ଟେଟ୍ରାନ୍ୟୁକ୍ଲିଏଟ୍ ଅର୍ କ୍ୱାଡ୍ରିନ୍ୟୁକ୍ଲିଏଟ୍। ୟୁନିନ୍ୟୁକ୍ଲିଏଟ୍ ହେଉଛି ପ୍ରଥମ ତିଆରି ହବ, ଗୋଟିଏ ନ୍ୟୁକ୍ଲିୟସ୍ ରହିଛି। ଇଟ୍ ଭ୍ୟାରିଜ୍ ଇନ୍ ସାଇଜ୍ 6 to 9, ସାଇଟୋପ୍ଲାଜମ ଇଜ୍ କ୍ଲିୟର, ଗ୍ଲାଇକୋଜେନ୍ ମାସ୍ ରହିଛି, ବଟ୍ ରଡ୍ ଲାଇକ୍ ବଡିଜ୍ which are called chromatoidal bodies, ତାର ଫଙ୍କସନ୍ ଜଣା ନାହିଁ ଏପର୍ଯ୍ୟନ୍ତ, କିନ୍ତୁ କ୍ରୋମାଟୋଡେଲ କାହିଁ କୁହାଗଲା, ବିକଜ ଇଟ୍ ଷ୍ଟେନ୍ସ, ଓକେ, ଇଟ୍ ଷ୍ଟେନ୍ସ, କ୍ରୋମା ମିନ୍ସ କଲର ନେଇ ପାରୁଛି, and can be also seen. So these are large, smooth oblong structures, kota chromatoidal bodies. I mean, dekhiba our next slide re. It may be one or more in number with rounded ends. Then this uninucleate turns into the tara nucleus cable divide karijibo. The nucleus of uninucleate divides to form a binucleate cyst with two nuclei. It retains all other characters of uninucleate cyst. Binucleate cyst gradually transforms into quadrinucleate or tetranucleate condition. So here you can see the chromatoidal bars, here one to several oblong rods with blunt ends or round ends are found. The function is yet to be determined. So this is the glycogen mass. They are associated with this glycogen mass. A single nucleus is present. You can see here, this is the stained binucleate cyst. You can see here binucleate cyst of the, so dita dekho nucleus eti dekhagala, dekho you can see here the two nucleus, it is diagrammatic, and again these chromatoidal bars are present, which are in variable number. The glycogen mass is also present. This is binucleate stage. This is the quadrinucleate or tetranucleate stage. It is stained, okay, it is not diagrammatic, it is stained. You can clearly see these four nucleus present in the cyst. Four nucleus, chatta jako nucleus dekha jauchi. Then the diagrammatic form of these four nucleus. So quadrinucleate cyst is the infective stage. In this stage, when if they come out, they are capable for infection, fresh infection. So quadrinucleate cyst has a small nucleus because of successive mitotic division, but retains all the characteristic features of the trophozoite. Then this is the life cycle of Entamoeba histolytica. This is the trophozoite stage. After entry, divides, binary fission, increase their number. These again become feeding stage, go on increasing their number, and finally some continue their division, and others become restricted for division, and they prepare themselves to undergo cystation. So encystment occurs due to the extrudation of the food materials, and they become almost oval in shape, and in during cyst formation they pass through pre-cystic stage. Then cyst is formed here. This is the pre-cystic stage. Cyst is formed. Initially it is uninucleate, becomes binucleate, tetranucleate, and in this condition it enters through contaminated food or drink, where again each of them divide once, all these four nucleus divide once to form 8 metacystic trophozoite, which are the infective stage. They divide and they are the infective stage. Here you can see these how these mature colored Roichi in the colored stage. So coming to the pathogenesis, the symptoms, mode of transmission, definitely eating raw vegetables or fruits which have been contaminated with uh, the parasite, then drinking cyst contaminated water. Mechanical vectors also play a greater role, like houseflies and cockroach. Food uncovered, personal unhygienic habits, personal unhygienic habits. Just a minute. And then poor sanitary condition. It has been proposed that sexually transmission of histolytic has been also been reported in some people. Regarding pathogenesis, one is non-invasive pathogenesis, means you host the parasite but you don't show any symptoms, and one is invasive trophozoite. You have the parasite, you show certain symptoms in the form of sufferings from these organs like liver, spleen, and lungs. They are affected. They are called symptomatic amoebiasis, means you present certain symptoms, abdominal pain ho ba cramps ho, loose jo dysentery ho. So sometimes amoebic ulcer in the mucosa. Ulcer means মানে जो histolytic proteolytic enzymes release करती, they dissolve the tissue lining the intestine and gain entry into this. So ସେଥିରୁ blood ବାହାରିବା, that is why bloody stool comes out. And sometimes the toxin also they are sprayed from to different parts of the body. The infection ଟା ବହୁତ ଅଧିକ ହେଇଯାଇଥିବ ଆଉ that is called colitis. Okay. Term is called ulcerative colitis or abdominal colitis. It infects the colon region. Colitis. Ulcer may be confined to the cloacal region. Ulcer means ଘା ହେଇଯିବ ବା କଣା ହେଇଯିବ, ସେଥିରୁ ରକ୍ତ ବାହାରିବ. And non-dysenteric amoebic colitis. Amoebic colitis, it is uncomplicated type of amoebiasis using usually chronic in nature. Symptoms include in intermittent diarrhea. ତା ମାନେ non symptomatic, ଆଜି ହେଇଗଲା କିଛି ଦିନ ପରେ ପୁଣି diarrhea ହେଇଯିବ, mucus in the stool ରେ mucus ଆସିବ, ଲାଳ ପଡିବ, abdominal cramps and flatulence. Patients might have slight inflammation in the colon with a few amoebic ulcers. So this is called non-dysentery. Rarely dysentery may. Then acute amoebic dysentery by bloody diarrhea, frequent passage of stool, and bloody stools are present. Bloody stool মানে ତା ଦେହରେ ରକ୍ତ ପଡେ ଆଉ ରକ୍ତଟା ଆସିଥାଏ. Depending upon the amount of blood present, you can determine what is the nature of the infection or the nature of the ulcer. Chronic amoebic dysentery, amoebic colitis ପୁରା ଛିଦ୍ର ହେଇଯିବ, rupture ହେଇଯିବ. So people taking corticosteroids are also susceptible to amoebic colitis. Amoebic appendicitis, that means if they harbor the appendix, they will inflame. Our inflammation ହେଇଗଲା ପରେ, if the appendix rupture, then the entire toxin will spread throughout the body. ତା ପୂର୍ବରୁ ଏତେ pain ହବ, it immediately it necessitates the surgical removal of the appendix by means of appendicitis. ତା ଛଡା ଯୋଉ ଏକ୍ସଟ୍ରା ଇଣ୍ଟେଷ୍ଟାଇନାଲ ରିଜନ ଯୋଉଠି ସେମାନେ ତ ମୁଭମେଣ୍ଟ କରୁଛନ୍ତି ବ୍ଲଡରେ, ହେପାଟିକ ଆମିବାସିସ୍ with the liver abscess formation of a liver ulceration. Abscess means fluid deposit ହେଇଯିବ in certain region of the liver, ତାକୁ କୁହାଯାଏ liver abscess, by their cytolytic action causing necrosis by death of the liver cells. Then splenic amoebiasis, if they infect the spleen also in like liver abscess, splenic abscess ତାକୁ କୁହାଯାଏ, সেটা ମଧ୍ୟ ଦେଖାଯିବ. Pulmonary amoebiasis means if they infect the lungs, so that is called primary pulmonary amoebiasis. If they reach the lungs by the pulmonary capillaries. So pulmonary amoebiasis ରେ, pulmonary amoebiasis ମାନେ କ'ଣ, if they infect the lungs infection ପରେ କ'ଣ ହବ, the symptoms are fever, chest pain, severe chest pain extending up to the right shoulder, dyspnea left shoulder re chest pain hele seta related to the cardiac non function. Right shoulder re ye hele chest pain re, that is something wrong with the lungs. Then dyspnea breathlessness rahibo because surface area kami jibo for the exchange of the respiratory gases. Dry cough sukhila kaso hobo, dry cough without mucus. Then cerebral amoebasis means if the reaches the brain. Uh, but it is very rare. Trophozoite gain entry into the brain, and it has যদি brain রে ସେମାନେ ରହିଗଲେ ଆଉ brain ରେ abscess ହେଇଗଲା, mortality ନିଶ୍ଚିତ, high mortality 90%. If they reach the brain, 90% cases रे मरी जाती, and the person may die within 12 to 72 hours. That is why it is not advisable to go through any prone any activities which is prone to infection with this entamoeba histolytica. Cutaneous amoebiasis means due to the puncture of amoebic abscess or of surgical wound infected with entamoeba. Surgical wound means we were dressing if it is infected, তো সেটা হচ্ছে cutaneous বা skin amoebiasis. So I think we have already covered it. Diagnosis, সদাবেলে the stool is examined to find silvery shining silvery globular structures present in the stool. তো সেটা stool examination ଆମେ ଜାଣିପାରିବା. ତାପରେ serology and others. So I think today, tomorrow will be our last class. I'll try to complete the remaining syllabus within the stipulated time. And as I've told you, I will share the slides with you. So thank you, all of you.
Thank you, sir.
Okay, we'll meet tomorrow.
Thank you, sir.